antibiotic resistance. I think that's my plan. All right, so let's get started. So we're going to talk about some of the research we are doing in my lab. So I'm going to present some of those, but I will start with probiotic. Again, the probiotic is the application. We've been talking about biofilm produced by probiotic or lactic acid bacteria, and I already the day one I gave you some examples how probiotics are or lactic acid bacteria can form biofilm and their implication in industrial setting or health setup. So I just listed few lactic acid bacteria and probiotics. These are pretty well known: Bifidobacterium, Lactobacillus, Lactococcus, Enterococcus, Pediococcus, and yeast Saccharomyces, just to name few of them. But then if you look at the panel of probiotic or lactic acid bacteria we use, there are different groups. Um, so the one of the deleterious or negative effect of biofilm produced by lactic acid bacteria could be excess accumulation of metabolic byproducts. A lot of times involved in food spoilage. I had a lot of slides, actually I cut it down, I didn't want to bring a lot of information. They can form off flavor, or they could also produce some amines. You heard about amines, small amines or biogenic amines. Sometimes some of those amines could be have negative effect, sometimes more allergic effect, like histamine example. It's a breakdown product of the amino acids. Serine, cadaverine, a lot of those small amines, and it's because of the breakdown by a lot of lactic acid bacteria. Even in cheese, cheese products, sometimes you eat certain cheese and then you start showing allergic response. That's because of the breakdown of some of those proteins by bacteria to form amines. So those are the negative impact. And then of course we talked about the biofilm can be the place where pathogens can harbor or can hide, right? And in this case, even though produced by lactic acid or probiotics, and that can be source as well. What about the beneficial effect? That's what we're going to talk today, right? So beneficial effect, it can have a protective effect for food fermentation, right? It can help in the food fermentation. I showed you some data last time. And also they can produce antimicrobial agents, whether it's acid or bacteriocin-like molecules. Those byproducts can inhibit pathogens. And also health attributes, right? So we're going to talk about mostly the positive, the beneficial effect of probiotics today. Again, I'm going to review some of those things you have already seen. We talked about the gut barrier, right? We went through natural or innate immune system, what we have in our gut, right? Starting with the mouth, with the saliva, stomach acid, then small intestinal antimicrobial compounds including bile, peptides, and natural microflora, and also some immune cells, right? They provide protection. We already discussed that yesterday. So that barrier can prevent pathogen from transferring through the GI tract, as well as also the lactic acid bacteria, including probiotics. They have to also overcome these innate defenses so that they could colonize in the gut and can have positive impact. What about the physical barrier? We talked about that again, right? Let's review it briefly. So we are looking at the gastrointestinal barrier, and we are talking about the physical lining of epithelial cells. So these are the villi structure in the gut, and if you look closely, you have seen already these are the villi structure, right? And these are the crypt, and these white locations are the goblet cells secreting mucus, right? And this is kind of an example of healthy ileal villi structure, and these you will see also the mucus, okay? And these are the crypt where the epithelial cells origin, and they migrate upwards as the cell mature and die, okay? And if you look closely, then you can see this epithelial cell lining. If you take two epithelial cells, and here is one, and this is another one, and this is the cell. And this tiny junction is called tight junction, right? This is the tight junction between two cells. And this is the microvilli structure, filamentous structure. Now go deeper into the cellular architecture, then you will see this area only. And if we magnify it further, you can see this little area. These are the microvilli. This is called tight junction. Going over that, this is adherence junction. This is desmosome. This is gap junction. This is how the epithelial cell line up, right? And if you look closely, what are those proteins? We went over that a little bit. You see these tight junction proteins are claudin, occludin, zonocludin, F-actin, and then the myosin light chain kinase. These are the components of the tight junction, right? And then the adherent junction, the major protein is E-cadherin. Then also you have alpha catenin, beta catenin. These are the proteins holding this junction together, right? And then this, you have also seen this, the schematics, how these proteins are held together in the tight junction or adherent junction as well as the desmosome. Then as I mentioned that also you can stain individual proteins to assess the epithelial integrity by measuring the protein level. So this is confocal imaging showing you the presence of zona occludin, and these are the E-cadherin, which are the long proteins. If these E-cadherin proteins are destabilized or broken down or degraded, you will not be able to see these proteins, right? This disappears. This is the way we can monitor the integrity of the junction, epithelial tight junction. What else I need you to know? If we talked about few times about the leaky gut, right? The epithelial permeability, if it enhances due to infection or due to heat, due to any abnormal conditions, you experience leaky gut, right? And the level of the pore formation, literally you can measure the level, how the permeability occurs. So if it is a pore pathway, which is very small, about 6 to 8 angstrom, that means the small molecules can pass through from the lumen to the subcellular spaces. If the epithelial barrier disruption is about 100 angstrom, and we call it leak pathway. And that's when the water and the other molecules start to leak out. And the best way to assess that, you can measure the flow of two different types of molecules. And FD4 implies actually fluorescent FITC conjugated dextran molecule, about 4 kilodalton, and this is 40 kilodalton size molecule. So what we do, we measure the transfer of molecules from the upper layer in the transwell setup when you grow the epithelial lining, and then you look for that molecule in the basal layer, right? If there is a tight junction compromise, this small molecule would go through, and based on the size of the molecule passes through, you can calculate the pore size or the level of disruption or barrier damage. And on the transwell setup cell culture model, you can definitely calculate that, as well as bacterial translocation. And the unrestricted pathway is the one when your tight junction is totally wiped out. Means sometimes the damage happens so severe, the bacteria can easily pass through. So the larger molecules, bacteria, they all pass through. So if you have a diarrheal situation like Shigella dysentery or Salmonella or Campylobacter or even Helicobacter, or Vibrio, sometimes they cause severe damage. The bacteria also can pass through. So these are the level of epithelial barrier damage or dysfunction you can measure. And in the animal model, how do you assess it? We need to feed the animal about 4 to 5 hours before you sacrifice it. Okay? Because a lot of our data I will show you, we measure that in the mouse, and you orally gavage them 4 to 5 hours before sacrifice. And most of the time we use FD4 because you try to measure the leak pathway. That's where we are interested. But you can measure also FD40 or FD70. These are the larger molecule. And if they pass through, that means you can assess your damage is severe. So that means depending on the agent, whether it's a toxin or any abnormal conditions in the gut, you can literally measure the level of barrier disruption you have. Okay, that's very important, because if you have a severe damage, then repair would take longer, right? Because you always want to repair our gut barrier dysfunction so that you can heal fast, and that's the part of the gut health. And so after we sacrifice, we collect this serum and also urine, and we monitor this molecule in these two fluid, whether it's in the serum or urine. So if you see high level of this molecule in serum or urine, then you know you got a severe gut problem. Okay. In human also, if you go and visit gastroenterologists, they will give you some drink and let you, drink that. After that, you can collect it in the serum, mostly in the urine. They will measure it, and if you have a high level, that means you got a gut problem. Okay. All right. So this is how we're going to use it in our experimental approach. And a little bit more, I want to tell you. And if this damage happens in the tight junction, and this is also called paracellular migration. So that means microorganism, they can migrate through paracellular route from lumen to basolateral side, in the below the epithelial cell, and then it gets to the blood circulation. Okay. Some microbes actually they produce enzyme, and this enzyme also can break down this protein, and so it opens up the path. Okay. And some cases the organism can activate actin skeletal, means the cellular protein structure which maintains the integrity of those cells, and if you disrupt that actin molecule, and sometimes this junction open up. So these are the varieties of ways microorganisms manipulate the cellular function so that they can get inside. Okay. Again, look at it from the pathogen side. This is what we are looking at. This is another way. So one of the, again, you saw this picture before. What is the signaling cascade that leads to opening of this tight junction? Okay. Sometimes, like we talked about LPAs, peptidoglycan, right? Those gut-derived microbial components, and they are highly inflammatory, right? They can also cause tight junction barrier dysfunction. What they do, if they are binding to a receptor, and then this MyD88 pathway, they can activate NF-kappa B. Okay, and we talked about that NF-kappa B signaling cascade, and this P55 or P65, they're all actually in a normal state. They're forming a cluster, and when they're activated, they separate and they get inside the nucleus. The first thing they will do, they will bind to the promoter, and then they will transcribe this, and then from messenger RNA, and the result is, outcome is inflammatory cytokines like TNF alpha, IL-1, IL-6. These are the inflammatory cytokines are produced. So that's the indication that inflammation is taking place. And this low level of cytokine we talked yesterday, it's good for your health from the standpoint of activating immune cells. Okay, and if this happens severe, and then it come back also, it can also regulate this tight junction barrier. So it also make it leaky gut. Okay, and one of the other protein I mentioned is that myosin light chain kinase, and this kinase enzyme controls this tight junction protein. So that means this tight junction protein is holding together. If the enzyme get activated, these proteins are pulled from the junction area. Are you following me? So these proteins which are holding together, I showed you before, and this tight junction is kind of like a sewing, right, with the thread, and this enzyme is get phosphorylated, MLC2 phosphorylated myosin light chain, and then actually it controls these proteins which are holding the junction together. So if I'm a microbe, or if I'm the stimulant, I can activate this enzyme, and then what it does, it pulls all the proteins away from the junction and open the gut barrier. The epithelial become leaky. Are you following me? So that's when the molecule from the lumen can get in, or the molecule fluid from inside can get out. So that's the during diarrheal condition. Okay. So this is one of those established mechanisms used by at least some of these molecules. And this is just showing that again in the cartoon form, showing that these proteins, how they are regulated, and they can pull away. And usually we call it, these proteins recycle, means it's gone inside the cytoplasm, and then few minutes later it comes back. So it's not an irreversible process, it's a reversible process. You can make it irreversible if it is a severe damage happens. It takes longer to repair it. Okay? So this is normally continuously happening in the body. So if you are heat stressed, okay, this can open up, that's when you show signs of diarrhea or vomiting. Okay? So that's an example. If you want to learn more about it, the several different microorganisms, this is, we wrote a review years ago, and you can see all those enteric pathogens listed here. They use certain virulence factor, and they also target a lot of these proteins or signaling pathway to open the tight junction barrier. So that's how they get easy access to our intestinal mucosal layer, and also distant organs. Okay, and we also mentioned about the leaky gut, right? Look at this picture. You saw this before, right? Whether it's a pathogens or any natural organisms in the colon, colonic tissues, or in the gut, they can cause inflammation, right? Inflamed epithelial barrier damage. Also colonization of opportunistic pathogens. Here's a Staph aureus. Microbial dysbiosis, right? All of those conditions can cause leaky gut. So therefore, this microorganism is going to come below the epithelial lining and then activate immune cells. As I said before, that you don't want excessive activation of immune cells, and they cause cytokine production, exacerbate the inflammatory process. Okay, and then also we talked about the consequence of having leaky gut, right? And also all those molecules getting in, then all the cytokines and the molecules circulating in the body. So whether from lungs or gut, you end up getting into the brain, right? And also the joint. We talked about arthritis, right? That's where the antigen antibody complex or some of the cytokines accumulates, cause inflammation or swelling. And then also the gut brain axis. Okay, so as I also said before, that leaky gut leads to number of different diseases. So it's a serious consequence or consequential disease form. So allergic or autoimmune conditions, asthma, atopic dermatitis, allergic rhinitis, celiac disease, inflammatory bowel disease. So a lot of those diseases linked to leaky gut. Also I mentioned that like diabetes, obesity, rheumatoid arthritis, multiple sclerosis, autoimmune hepatitis. So there are a number of diseases linked to leaky gut. Alzheimer's, Parkinson's, chronic depression, autism. So a lot of those diseases are now may have started from the gut. Okay, so leaky gut is very important. So how do we repair all of those things? So that's where I'm going to talk about the probiotic and what we did in our research to help mitigate some of those gut related complications that to repair the leaky gut. I don't need to spend time on defining what is probiotics, prebiotics, symbiotics. I presume all of you know it. I just want to point out only a few things about probiotics. So there are some consensus people, the experts, they met and they are trying to redefine what is actually probiotic. Sometimes it gets really confused. So if you see any lactic acid bacteria, they used for fermentation of product, do you call them probiotic? By definition, no. One way you can call it probiotic as long as you can demonstrate that it has a health beneficial effect. Okay, so that's the key word, health benefit. Okay, if they don't have a health benefit, direct health benefit, not indirect one, and then you cannot really call them probiotic. Other aspect is that you know this massive microbiome, microbiota research we are doing, right? The whole purpose of this is that if you know or not, why we are studying microbiota, microbial community. At the end of the day, we want some microorganisms, we can use them as probiotics. Okay, if you can identify whether it's for skin disease or heart disease or gut ailment, if we could find or identify some organisms from this unknown community, we want to use them as a probiotic eventually. Okay, and that's where we are heading. And so this panel decided that can you call this microbial community as probiotic? Can you call this microbial community you are now studying and you are saying that this number of organisms have certain impact on your health? Okay. The question is, can you call them probiotic? Because a lot of times people may claim that, oh, I got this from this community and I can prove that these are probiotic. Can you claim them as a probiotic? So the answer is, I would say no, unless you define those cultures clearly. Genetic information as to genetic, what taxa they are, what their function, every detail of them. Unless you can do that, then you don't have any claim to establish. So they are trying to warn you that if you have a microbiota transplant, fecal microbiota transplant, right, you cannot call them probiotic because it changes. You have no clue what are the population, you have general idea, but you may not know every detail of those organisms. Okay, like we know about lactobacillus acidophilus, lactobacillus rhamnosus, or bifidobacterium. Those are known probiotics. So that's the warning they are trying to give you. So keep that in mind as you as you study. Then also we use a lot of definition for probiotic, right? So not only probiotic, postbiotic, pharmabiotics, next generation probiotic, all kinds of names there. I want you to again familiar with that, okay, where we are going with this probiotic research. And then also there are some known organisms I already mentioned. Some of those unusual ones like E. coli, they use that Nisle strain as a probiotic. And then spore forming bacteria like bacillus, they are also very good probiotic. Do you have any idea why people like to use spore formers? Spore former probiotic? Any idea? Spore formers, they can withstand heat. They can withstand a lot of processing conditions, right? So that's why it's easy to use it in a product because they have a self-stable, self-life, right? If you are formulating a feed, and that can have a lot of temperature abuse, right? If you have live organism as a supplement, they may die. But if you have a spore, they will survive. And as soon as they get into your GI tract, they can become vegetative cell. Now they can exert their probiotic effect. So that's why a lot of company, they like that the spore former for practical application. Sometimes even some people try to add it to a cake mix. Think about it. Cake mix, they will add spore former. And so during baking, with the idea that they are going to convert into vegetative cell. And that's when they can have a probiotic action. If you use just the regular lactobacillus or other vegetative organism, they are not going to survive. So these are some of the clues, remember those. And this is in US, okay. One night I sat down in the computer and tried to find out how many different probiotics are available. It's huge, enormous. It is growing now. I mean, I know it's in the Eastern culture, people here in the Asian countries, people use a lot of probiotics, right? And there are a lot of traditional use from generation after generation. In the Western culture, especially in the North America, this is beginning to gain momentum. People started to believing probiotics has positive effect, right? They never believed that probiotic has any good effect. So now once they understood, now a lot of companies try to make money out of it, right? So they will market all of those, a lot of these are similar organisms, maybe same organism. They use multiple strains of same organism. It's kind of ridiculous how they market it and they sell it. And you go to pharmacy or grocery store or any health stores, you'll find a lot of these probiotics in the shelf. So I think I kind of raised questions that do they work? A lot of those questions I'll try to address that. But a lot of times this is a feel good, right? A probiotic, it has a positive effect and they sell. And also for the pets, right? Pets also is a huge market. The cats, dogs, that's very big. And of course the livestock also. But these are all actually for humans. And of course the symbiotic, I don't want to again spend time. There was a review by the consensus panel. They talked about symbiotic. They came up with certain criteria to establish a symbiotic. Just the two definitions I want to point it out. Complementary symbiotic. So the two symbiotic with the probiotic, if you mix it, they complement each other. They don't compete. And the other one is called synergistic. So like if you have a symbiotic, probiotic, the action should be added. You see enhanced effect. We are doing one research with our probiotics with blueberry polyphenol. We noticed that if the probiotic has one effect, the polyphenol has effect, and if you add two of them together, you see right here. And that's a true example of synergistic effect. Okay, just to point out. Again, those the references are there to follow. This actually is this most important slide, or there's another one I will talk about. I tried to cover a lot of these things yesterday. Due to lack of time, I didn't get enough time to talk about the immune response, right? So we talked about food antigen, right? You eat food naturally and how your body responds to it, what immune system does. And we discussed also that we don't develop immunity against food, right? Then we will not survive. Food is a foreign antigen, right? And if you have an immune system against food, it's going to be a problem, right? But we want an immune system against pathogens. So just to, I don't know whether we'll be able to focus so much on this left side, food antigen, right? So we have a healthy condition, mucus, antimicrobial peptide, and then IgA, right? And then what you look at this immune response, okay, it's fascinating that all those this TGF beta, IL-10, IL-22, a lot of these are upregulated. And the type of cells, immune cells, I started talking a little bit, regulatory T cell. This is also another one, Rho gamma T. This is also T lymphocytes, macrophage B cells, and CD103. This is actually dendritic cells. They have a positive impact, okay? And the antigen processing we talked about, right? Antigen presentation, and they have a greater tolerance for it. They don't present to the immune cells. So therefore, the immune cells don't, or T cells, don't think that they are foreign. So that's why they are not going to induce immune system. And if you look at the mucus goes up, AMP up, IgA up, PR pattern recognition receptors are up. But if you look at the pathogen, the dysbiotic condition, the disease condition, right? Those are all negative. See this going down here. Look at the type of cytokines goes up. These are all inflammatory cytokines. And these are the clear marker. The type of T cells also respond. These are clear marker that you are under dysbiotic conditions where your immune system is very weak, very poor. They are showing inflammatory response. Do you see the balance? And this balance always playing a role how when you take food or probiotic or healthy diet, okay, healthy conditions, versus if you have a dysbiotic or poor food habit, like a Western diet. We talk about hamburger, pizza, all kinds of nasty, the sugary food, all kinds of stuff have fall under this category. So all negative effects. And the healthy diet is fall in this side. And so the immune system works very positively in that side. So keep that picture in your head. And the other one, again, is pretty complex. I would recommend you to go through this article published in Nature Medicine. And this essentially summarizes how some of those probiotics positively impact health. Okay? If I can just quickly tell you that they produce short chain fatty acids, and they also the forming biofilm is one of the criteria. This is the community. And remember, I talked to you about that can slightly cause inflammatory response through LTA. I think we had a discussion yesterday. And also with the shortage dependent pili we had talked about yesterday. So they can also activate these epithelial cells. Okay? And then also the epithelial dendritic cells bring those antigens or probiotics inside, and it also initiate this regulatory T cell pathway, which are the immunomodulatory response. And there also you will see the anti-inflammatory response, and also the tight junction. The probiotic, it maintains the tight junction. It works opposite to pathogen. If pathogens open it, probiotics will close it. And this is again, you see upregulation of claudin, zona occludin protein, mucin, MUC2 I mentioned, their level also goes up. So these are all positive attributes due to probiotic. And also GABA. I think some of you are working GABA and some of these probiotics, and these are also secreted, and it also directly affects the brain. Okay? And then also the other functions. So if you go through this review, I think it did a great job summarizing those immunomodulatory, anti-inflammatory, GABA, all of this response, right? So let me show some of our data, okay? And I think I set up the base, the background well, so you can follow this research. And so this is a company, small company gave us some money, and they market for probiotic blend for their pet food. They have a dog feed they sell, and they gave us this probiotic to do some testing, asking us how well this works. And so Nick, my grad student, he started studying a little bit on that one. So what we did, this blend actually has these three organisms, pretty recognized organism, right? Enterococcus, lactobacillus, lactic acidibacillus, KGI. And so few things we did, very simple study, just to show you what property you are looking at. Because it is for dog, so we had to use canine cell model, which is the kidney cell. And here we looked at the pathogen inactivation, biofilm formation. As you recognize, biofilm characteristic is one of the important characteristics for probiotics to have. We recognized that a while ago, and many people now recognize that. And then also we looked at anti-inflammatory response. And first, I think it's very simple. You measure the activity, right? If they work against some of those pathogens, common pathogens. So what you noticed actually, the bacteria has that antimicrobial effect. but not the supernatant. If you don't see the activity in the supernatant, clearly says that probably these bacteriocin-like molecules are not quite involved. So we haven't really want to explore what is causing it. That was not the purpose. We just wanted to screen with a limited amount of funding, so we just left it there. But most importantly, we wanted to see, do they form biofilm? If we recognize biofilm formation is one of the positive traits or attributes, so we want to measure that. So we compared that with Staph aureus. In 48 hours, you can see Staph aureus form a strong biofilm. But whereas my lab max 3 did not quite form that, and LKGI also pretty weak biofilm. But after 72 hours, Staph aureus remained pretty same or similar, but these two other ones picked it up. So that means it's time dependent biofilm formation. They take longer, okay? And just to tell you that how long, make sure the animals are exposed or how often you have to feed them so that it's maintained certain level, right? In order to get the positive attribute. And by counting colonies, this is by crystal violet staining, by colony counting also you can prove the same information. And then you can also stain it by Giemsa stain. If you take the biofilm, and here you can see this the bacterial biofilm by these three organisms, and you can see clearly the strong biofilm formation. Another thing I also mentioned that the morphology may change, right, in the biofilm producing cells, and we did indeed notice the changes in the cell morphology in those organisms. Anyway, and then of course we wanted to see the adhesion to the kidney cell line, and you can see the LabMax 3. What we did actually in this case, we treated with LPS just to make the cell simulate the gut environment where your epithelial cells would be exposed to LPS, peptidoglycan, a lot of those molecules coming out of the bacteria. And so we wanted to see how well this works. And so naturally, if you look at all of those data together, you see this LabMax 3 form nice, impactful colonization on the MDCK cell line, which is the canine cell line, and compared to KGI produced a little bit more. And so they adhere, they form cluster like biofilm-like structure. And they are also non-toxic. And by LDH assay, you can confirm that these cells are not toxic. These probiotics are not causing cell damage. Okay, and then of course we looked at the cytokine profile. Okay, and in the cytokine profiling, and definitely one of the desirable cytokine we wanted is the IL-10. But the most importantly, the ratio between IL-10, which is anti-inflammatory, and TNF-alpha, which is inflammatory. So you look at this, the ratio is pretty low, so indicating that LabMax 3 is working moderately well, as could be a positive one like a lactobacillus KGI. Anyway, so we had some indication that it does immunomodulatory or anti-inflammatory response. Okay, so now I wanted to get into the what bioengineering strategy we took to help improve these probiotics to work against some diseases. Okay, so in this case, we wanted to see why some probiotics don't work. Okay, I'm trying to ask this basic question. Like a lot of times you take probiotics, it works. Sometimes it doesn't show positive effect, right? And that's where we are concerned about. So let's ask this question. So the probiotic organisms, you'll find them embedded in mucus layer. Okay, they never enclose to epithelial cells. This is under healthy condition. What happens if you are under disease condition or dysbiotic condition? So what dysbiotic condition looks like, your mucus layer thins out. We saw that, right? And then epithelial barrier also disrupted, right? And in that conditions, this is when you want your lactobacillus to be around, right? If they are probiotic, but they are the one which leave the body. So if they are not there, because the condition is not favorable, and they will disappear, therefore you will not have any positive effect, right? And that's the problem, right? So if you have leaky gut, we talked about inflammatory diseases, if it is in the animals, poor health, weight loss, disease susceptibility, survivability, and economic loss if it's for the livestock or farm animal. Okay, for human, we already know what consequences. So the question is, can you reverse this process? Okay, and reversing the process requires some diligence from your side, the scientific discovery. So one of the things we are looking at, what is the solution, right? And so the question is, what happens during dysbiotic condition? What do we know about the dysbiosis? why these lactobacillus are not staying in the gut when they should be, right? What can we do to keep them? So one of the things we noticed that heat shock protein 60 is one of the molecules that expressed at high level in the disease cells. Everybody with me? Okay, so that's one of those markers we are trying to work on. So what is HSP60? Okay, I'm going to explore a little bit more. So during infectious disease, it increases. Also inflammatory disease, metastatic cancer, cardiovascular disease, this molecule goes up. This is one of the greatest indicators. We are exploring some. This is not much data available. We are doing some work to understand why this molecule goes up. So we have a protein that binds to HSP60, okay, and that protein is called Listeria adhesion protein, or ADHE, which is called acetaldehyde alcohol dehydrogenase. Okay, this is one of those enzyme or protein. It's secreted by many organisms. Okay, and so what we wanted to do, if you can create a probiotic, are you following me? This is where the most important part coming in. We want to express this probiotic organism with this lab, okay, and this lab is coming from Listeria. This is where we worked on in our last 20-30 years, and so we figure out if we could express lab which reacts on HSP60, can we have this probiotic have a strong binding to those epithelial cells which are under stress, which are showing the signs of disease condition. And this is what we wanted to do. Okay, so HSP60 briefly, if you don't know about it, it's called chaperone protein. If you know, chaperone is that it deals with the protein synthesis, protein misfolding. They will remove those things from the cell, and also they have an immune lighting function. Some people claim, or we also claim, that immune signaling molecule, and it's a disease marker. Already I mentioned that. And so if you look at the cancer cells, they are finding out the HSP60 playing an important role for any kind of metastasis. What we found out, actually, I mentioned about TLR molecule. This HSP60 likely binds to TLR4. It activates NF-kappa B, and so it controls a lot of aspects of the cellular physiology, even the cancer. Okay, and also in the cardiovascular disease, what we are finding out is that if you have a hypoxia condition, like a lack of oxygen, and you are under stress, so HSP60 would be produced at high level, and some of those HSP60 is secreted, and also it can bind back to TLR4 I mentioned a minute ago, and it binds, it activates this inflammatory pathway, NF-kappa B pathway. Then you see a lot of inflammatory cytokines, and that complicates your heart disease or heart attack. Okay, so these are like another mechanism to figure out what happens when you suffer from cardiovascular disease. But this is one interesting aspect. I just pointed out that HSP60 expressed on the membrane to serve as a marker or serve as a signal that you are under stress or you are under bad situation, and then it could be health risk. Anyway, so we are interested in Listeria monocytogenes pathogenesis in our lab, and we said that, well, under Listeria monocytogenes infection, HSP60 expression goes up, and so naturally in your gut you are going to see some HSP60. Okay, and you can clearly see this HSP60 expressed on the epithelial cell outside. And my student many years ago, she actually, Jennifer, she first reported that lab binds to HSP60. So HSP60 is the receptor. And then later Christine, she reported that under stressful condition, including Listeria infection, you see a high level of HSP60 expression. And later on, we also proved that in another research. So we know HSP60 play an important role in Listeria infection, or Listeria literally can bind to HSP60 during infection. So I'm going to tell you a little bit. And I already talked about Listeria. I'm not going to spend much time on that. It's a foodborne pathogen. I also talked about they can cause gastroenteritis, neurolisteriosis, or pregnant women complications, right? And so we know that it's a foodborne pathogen. It comes through food. It passes through the GI tract. It can get into the mesenteric lymph node, blood circulation, then eventually brain, and in the mother, it can cause the fetoplacental barrier, right? And the fetus doesn't have immune system. Therefore, it cannot defend against Listeria. And so the fetus are highly susceptible. So stillbirth, premature birth, fetal death is very common in listeriosis cases. And it happens number of times. In US, it's always like anytime you have listeriosis outbreak, some women get their problem with the fetal death or premature birth. So it's highly problematic in humans. Of course, in animals also it can cause disease. Originally it came from cow. It caused circling disease because it affects the brain, and sheep also have abortion. Okay, so it's a true example of zoonotic disease. Okay, but it's a human pathogen. So as I said, it has a lot of pathogenic factors to work with. But our research lab, we contributed to the understanding of Listeria pathogenesis. We discovered that Listeria adhesion protein, that's we discovered, which is alcohol acetaldehyde dehydrogenase. It binds to HSP60 and it activates signaling cascade that leads to infection. Okay, so just briefly, in order to understand the bioengineered probiotic, I want you to understand a little bit about this lab protein, which is acetaldehyde dehydrogenase, alcohol dehydrogenase, right? And this is part of actually glycolytic pathway, glycolytic pathway. Glucose converted into acetyl-CoA. All of you should know, right? Glycolytic pathway. And then acetyl-CoA under aerobic condition, it can go to TCA cycle, right? Again ATP, right? And if it is anaerobic condition, bacteria can harvest some energy, and which is ALDH can convert it to acetaldehyde, and alcohol dehydrogenase convert it into ethanol. Okay, so this enzyme is called bifunctional enzyme, and it's about 96 kilodalton. We call it moonlight protein or housekeeping metabolic enzyme. It has a dual function: enzymatic function when it's need, harvest energy. Other function is pathogenesis. Okay, that's the beauty of it. That's why you call it moonlight. You don't see moonlight all the time, right? In a month. So you have a cycle, right? And so that's why they once in a while they serve as a pathogenic factor. And other interesting aspect we learned, we find it in the pathogenic and non-pathogenic bacteria. And non-pathogenic means Listeria inaqua example. Agree with me? So why non-pathogenic using this protein, which is a pathogenic factor? So in non-pathogen, it doesn't have that attachment function. Okay, only in the pathogens, and it has a strong binding affinity to the HSP60. So what we learned actually, this is Kwang Pyo and Bala. They did quite a bit of work. Actually, Kwang Pyo did. He's now professor in Korea. He's doing quite a bit of work on biofilm research. Bala is working for Nestle. So they did some initial work. So one of the things I mentioned that this protein doesn't have a leader sequence. These are inside the cell. And I showed you how they are excreted outside before, right? So they, in this case, this is gram positive. They is called SEK pathway. I mentioned that SEK2. This protein, it binds to the SEK-YEG and then gets out. And then if it is outside, and if it is not bound to the surface, it doesn't have any function. So what happens in pathogens, it binds to a receptor, which actually Dong Si proved that, and he published that recently, last 23. And he said that this protein gets out and it finds a protein to lodge, whereas the non-pathogen, they don't have that receptor, and therefore it gets lost. So both pathogen, non-pathogen make this protein. Are you all with me? Listeria monocytogenes pathogen, it makes this protein. Listeria inaqua, non-pathogen, it also makes this protein. But inaqua, it doesn't have this anchor. Okay? Therefore, this protein gets lost. But this protein is identical, only except I think I would say 3 or 4 amino acids out of 866 amino acids. Anyway, those proteins are same. So we also recently tried to understand the lab HSP60 complex. We finished that work, it's submitted, and this is a cryo-EM. You heard about a little bit about yesterday, the structure part. Here we did cryo-EM to understand the lab protein, and it comes as a pentamer, tetramer, and also it acts as also dimer. It comes with varieties of structures. It's very fascinating. Anyway, no time to discuss more detail, but you can see this. This is HSP60. You can see this one, multi-dimer, and this small part of the lab actually binds to it. Okay, this is what we understood. But without spending much time, this is just the illustration showing the lab binds to this receptor, which we call it internalin B on Listeria cell surface, and then part of that lab binds to HSP60, which is host cell. Are you getting it? Listeria binds to this host cell before it causes the damage, and so that's the mechanism which already revealed and we submitted it. It's under review right now. So how does lab help in the infection process? Okay, you remember this picture? I showed you this one earlier, right? Yesterday or day before. And so here actually lab binds, it activates NF-kappa B pathway, it opens up the tight junction barrier and it passes in between. Okay, and then because it is an intracellular pathogen, this bacteria also can invade through cell by another pathway. Okay, and this is what we learned because activation of myosin light chain kinase removes the claudin, occludin, and ecadherin, and that's how it goes through. The other aspect is the part of the innate immune system, and so before immune system wakes up, it can infect the host cell. Okay, and so quickly, other aspect also you learn that it uses called caveolin mediated endocytosis. Again, we use a knockout mice to prove that caveolin also help in the translocation of the bacteria. Not have enough time to go through this cellular mechanism, but just to get the big picture. Okay, the cell junction opens up, bacteria pass through. What are the cell biology? Which proteins are involved in opening this junction, right? Because you have to understand if you can close it during the disease or the repair process or the healthy gut process, right? So that's where we are going to get in. This is just saying, again, understanding cellular biology, how this caveolin form a complex with the occludin, and this complex with the Listeria monocytogenes. This is a clear evidence. They form this called caveolar endocytosis pathway to go through the cell. Okay, all right. So this is the summary picture. Again, if you are interested, this was published last year in MBio. Rishi was taking the lead in this research. So can probiotics prevent listeriosis? That's the first question we asked. That's what we were waiting for. So one of the things we did actually, that's Oki and Ku actually, she worked on it, and this is the first time we ever worked on probiotics. We had no intention of working on probiotics ever. We were working on Listeria pathogenesis. But in order to prevent Listeria, all this knowledge we gained, can you use probiotics to prevent Listeria infection? That's when we got into probiotic research. So you can say our knowledge goes back to 12 to 15 years maximum. Okay. So we looked at Listeria monocytogenes. We used just the traditional lactobacillus, rhamnosus, acidophilus, paracasiae. None of them actually inhibited Listeria on a cell culture model. Okay. And I already said that before. Sometimes Listeria probiotic works, sometimes it doesn't. So we want to make it work. So what we did actually, we engineered this probiotic. And Rishi, actually Marianne took the lead, and then Rishi completed it. So they were co-authors. So just briefly explain to you what we did. So we took that lactobacillus KGI, right? And we took this lab from monocytogenes and Listeria inaqua. One is pathogen, one is non-pathogen. And I mentioned before these proteins are same, sequence-wise. So why not take it from both? Let's see if they can protect. So once you create it, so you express this protein with this expression system. And these are two bioengineered probiotics. Sometimes I call it BLP, bioengineered lactobacillus probiotic, or we call it NGP, next generation probiotic. So I have used this interchangeably. And so first thing you want to prove that these proteins are expressed on the surface. They did. Okay, so you can clearly see that. Whereas the vector or wild type, they don't have this protein expression. Okay, first thing you want to see is that do they attach to the epithelial cell, as you promised. Okay, so if you look at the wild type or vector, they have a minimal adhesion to KCO2 cells. But if you have a BLP, they have a higher adhesion, because this is the adhesion protein in Listeria. And do they invade? They don't invade the cells. That's a good news, right? Do they go through the paracellular route, translocation route? They don't. So that is very good information. Then next we look at, is it HSP60 specific? So one way to do that is that we created a KCO2 cell line which has a HSP60 knocked down and also overexpressing. So if there is a standard HSP60 expression, this bioengineered probiotics show increased adhesion and exclusion of Listeria monocytogenes. And in this case, overexpression, you have a higher binding, no HSP60 expression, no binding. So that's the HSP60 specific response. And then we wanted to prove that in the animal. See how well it worked, right? So we used mice and we used pregnant guinea pigs. And so whole idea being is that we fed them This is more like a prophylactic. So you take the probiotic. That's what we do, right? You eat it for 10 days, and then we challenge them with the pathogen orally, and then we kill the animals, and then look for the listeria in different organs and tissues, as I mentioned before, right? And we did that in mouse, normal, and this is pregnant guinea pig, because pregnancy is a high risk factor, right? So let's look at the video. So this is naive LM control. This animal extremely sick. It's 48 hours later, right? This animal is not moving. It's going to die pretty soon. This one received wild type probiotic, right? And they are challenged with LM, and you can see equally sensitive. It's also terminal stage. Let's look at these two on the right. One receiving probiotic made by LM lab, and this is Listeria inaqua lab. Are you with me so far? Okay. Now let's look at that animal. That's quite healthy, right? It's moving around. What about this one? This is even more active. The whole idea being is that this bioengineer we created, we challenged them with LM, now they can resist the infection. And when you look at the number, actually LD50, I mentioned lethal dose 50, we are getting about 92% protection. The high level of animals are protected from this infection. So now you may ask the question, how, why, what is the mechanism, right? We address all of those things. If you want to read that paper, it got tons of data. And so they have increased adhesion, colonization in the ileum, cecum, colon, all those bioengineered probiotics. The most importantly, we wanted to see, do they form biofilm? That's important, because how do the mechanism work? So I don't know if the lights are dim enough. So here actually wild type, they are mostly present in the mucus. They never goes close to the epithelial cell. Whereas the bioengineered one, they attach directly to the epithelial cell, both of them, and clearly show that they are binding to the epithelial cell. It's called epithelial cell contact. And that's very important, why this works. And if you count them, you can clearly see there are high number of those probiotics attached to this epithelial cell. Do they form biofilm? Next question. And this one clearly shows, you can see this, all this biofilm, including mucus, sitting right on the epithelial cells. Everybody see that? Wild type, because we don't have the right marker to treat it, so we don't know. And so here also is showing that there is a competition between Listeria monocytogenes and bioengineered probiotics when they are binding to the epithelial cell. There are some elegant experiments we did. No time to discuss that, but if you want to know more about it, I'll be happy to talk offline. So they maintain epithelial barrier integrity. We showed that, right? Do they do that? So again, use the cell culture model. If you treat with the Listeria monocytogenes, this opens up. The molecule goes through, or Listeria goes through. If you have a bioengineered probiotic, look at the FD4 movement. It decreases significantly. So if you have a leaky gut, now with the probiotic, you can prevent the leaky gut. You can repair it. What about in the mouse? You can see the exactly same result. So here is the LM showing the FD4 leakage in the both serum and urine. And now with the bioengineered probiotic, you can see already like a control. So that improved also in the urine. So that's saying that it does. What about the protein lab? I mentioned about those proteins, right? So I would set it up, myosin light chain kinase, PMLC, claudin, occludin, ecadherin. Look at those proteins. Look at the LM. LM caused severe damage. All those proteins like claudin, look at this, disrupted. This is control to follow this. Occludin, also you can see they are severely disrupted because of the LM. Ecadherin, also these long proteins all disappear, disintegrated. So that's saying there is this barrier damage. What about this probiotic now? So even wild type did not do anything. But when you have these two, you can clearly see this claudin restored and ecadherin restored, and all those other proteins, whatever it's supposed to be. So it kind of reversing the whole process, only because they are attaching each other. Okay, so that worked very well. How about the cytokine, inflammatory response, anti-inflammatory, TNF alpha low, IL-6 low. What about the regulatory T cells, which modulate your immune system? I mentioned that earlier, right? Treg cells. And they are, whether you have probiotic or not, it's working very well. And if you have a Listeria monocytogenes infection, then also it works exactly the same way. And this is a good sign that you are modulating immune system to a level where you can improve the health of these animals. Okay. What about pregnant guinea pig? Let's look at that. If it can prevent pregnancy-related complications, this is very important. So Valerie was involved in this project, and this is a pregnant guinea pig. And I don't know whether you can afford to do this experiment in a routine basis. Guess what guinea pig cost? This pregnant guinea pig. We bought several of them. So each of them cost anywhere $300 to $400. So calculate that in rupees. It's about 85 rupees per dollar. Okay. So to do this experiment, you really have to have a lot of money. So at least we spent $15,000 to buy just the guinea pig. So you have a statistically equivalent number to prove it. Anyway, let's see if this probiotic, these guinea pigs were fed with this probiotic. And literally they like it. We have to give them in the water. They will just go, and instead of drinking water, they will just drink the probiotic. And we don't want to physically abuse any of those animals, say force feed them. We don't do that. Just leave it there, they will drink at their pleasure. And so they've been doing that. Then we challenge them with listeria. Now, one thing we did, we wanted to do subclinical infection. Very low doses of infection, because we don't want to take a risk of getting abortion. Then we don't get the result. And so we did a low level of infection. So the mothers, if it happens, if you are susceptible to listeriosis, sometimes you may not show any symptom, but the fetus would show symptom. Okay, so it's like a asymptomatic disease in pregnant women. So we try to prove that. And so when you did the low infection, mother is actually infected. If you look at the liver, mesenteric lymph node, all of them were positive. Spleen positive, liver positive. So most of the mothers, they are all positive. Blood also, we see less. That's good news. Lungs also, this is bioengineered fed guinea pig, are less, almost zero. Right? Kidney also zero. Whereas the wild type did not have protection. Let's prove that in the placenta. What happens? This is where actually magic happens. If you look at the placenta, both bioengineered probiotic prevented listeria from getting to the placenta. Are you with me so far? Because placenta, next page is fetus, right? What about the fetus? We looked at the bacteria in the fetal liver, and so we didn't see much. And that's what we got excited that it worked. Of course, the brain, we didn't give enough time, so the brain takes more time to go through the fetal brain. So this data from fetal tissues, right? And so I'll show you the picture. Hopefully it's not going to bother you. This is naive mouse, the placenta, and they have a multiple fetus. Okay, and you have from 4 to 5 babies they carry. And so here, when you have a wild type probiotic LM, you can see all these fetus are dead, and you can see dark color. And if you have this bioengineered probiotic, in this case, we've been using actually Listeria inaqua lab expressing bioengineer, and you can see this healthy. And what about individual fetus level? This is naive. This is when it is infected by LM. You can see all bloody, and this color, look at this white, still natural color, and it's protected. So this gives you an idea that we are able to prevent feto-placental transmission of Listeria, right, from mother to fetus. So we'll be able to protect the fetus against listeriosis. So that's one of our drives, and this is our future goal. See if it can be used in the human application. And immunologically speaking, it did whatever is expected. Not to spend time. Also another study, I think I don't want to spend a lot of time, because I think we are very close to be done. And Nick also did that colitis model, and he showed real interesting results, and it shows the protection against the colitis. Again, you have a high level of HSP60 expression, and you can use this probiotic to prevent colitis. It works very well, and a lot of his data he showed. Again, like I said, in the interest of time, I don't want to spend a lot of time, but if you have a colitis or inflammation, and this probiotic is also going to work because of the similar mechanism. Okay? All right, so you can see that serum FD4 level went down because it repaired the leaky gut very well. So this is going to be a fantastic probiotic against any kind of leaky gut. That's what we are looking at. HSP60 expression also went down. You can clearly see here. All those other cases, you have a high level of HSP60 expression. And I want to show this video. So when you have this colitis, and if you feed these animals with this probiotic, you have about 89%, 90% survival rate. Whereas this DSS, which is the chemical-induced colitis, and it causes quite a bit of mortality. Let me show you this video. I think it will tell you the whole story. This animal, after they are treated with the DSS, you have a severe diarrhea, sodium feces, and then they started drinking this probiotic, right? You want to see them, they repair, they heal, right? And so those animals, actually this animal, this one, you see this one? Let me turn on the video. Let's see if it is, actually video is on. This animal is totally sitting there, not moving at all. And he had been on probiotic only day 4. He just recently received the probiotic. But it was in the dying stage, death stage. Let's look at the next one. So it's been already 6 days on the probiotic, that bioengineered probiotic. You can see this start moving, right? What about the next one? It's been on 8 days on probiotic. Same animal. It's over. We are looking at the progressively how they repair. And look at the one after 10 days of feeding probiotic. So you can see those people suffer from colitis or inflammatory bowel disease or severe damage. This probiotic can help because it binds to the receptor and then can make cell-to-cell contact and have a strong immunomodulatory response. We've got tons of data. We don't want to spend time, but showing that it works. We did microbiome also. I want to wrap it up. So every story I told you, this is my last slide, summary slide, saying that why and how it works. So essentially, if you have lactobacillus expressing these proteins, LAP, it can bind to the receptor. Okay, as soon as it makes the contact, whether you have a listeriosis or inflammatory bowel disease, it starts the magic, right? If it is competing with the Listeria monocytogenes, it prevents. So what you are seeing actually, it improves epithelial barrier function, right? So through myosin light chain kinase activity, pathogen opens it, and probiotic it closes. This is the opposite aspect. So that's why it is working very well. Anti-inflammatory response, all the cytokines, TNF alpha went down, IL-6 went down, and interferon gamma up, IL-10 up, TGF beta up. We want them. And then T regulatory function, Treg cell, and we saw this improvement significant. So it does immunomodulation. This is how it works. Okay. So we are not the only one doing bioengineering of probiotics. A lot of these are done for these specific pathogens. They are using kind of like a vaccine carrier. This is a very good system. A lot of work being done, or still people doing a lot of work. I want to leave you with a story from a lady. She went through pregnancy-related complications. She's from Indiana. Her name is Amanda Wright, and she keep writing me letters because she saw our research. She lost her first pregnancy due to listeria. Okay, and she's trying to be mother for the very first time, and first pregnancy went through complications, and she lost the fetus. And so once she lost it, then she saw our paper. We published this, and of course we had news release, and saying that if you have a listeriosis problem, and a lot of times mothers suffer from that, and she was totally devastated because she lost her baby. Then she said, well, I want to be mother, so second pregnancy. That's when she started writing me. She's asking me, can I have this probiotic? I said, no, you can't, because we don't have approval. It needs to be approved before I can give it to anybody. And she was so convinced, so she kept writing, and so she started writing several letters. So during her pregnancy, I was kind of like I never seen her. She just writes me. It's like a pen pal. And so I'm kind of like a counselor, trying to give her advice. She's afraid of eating any food. Think about it. If you are afraid of eating food, how are you going to survive? You're scared that, oh, listeria may be there in the dirt. Listeria may be there in the water. Listeria may be in the leafy green or fruit or salad or sandwich I'm eating. You can be eating a nightmare. And she went through that second pregnancy, and fortunately she was lucky. She had a healthy baby. Then, as I'm going to tell her story a little bit because she kept writing, and these are some of the language she used in her email. Just trying to emphasize, a lot of times we try to address one person at a time. This is how I see it, right? If I can help one person. So she said, Arun, I have a crippling fear of listeria during pregnancy. Recently miscarried, and I'm trying again to conceive. So that was the second time when she started writing. And she wrote a long letter. I just gave some highlights. The work you have done is so important and would truly save so many lives, not just from the actual infection itself, but also in terms of the mental anguish. Like I said, any food she wants to touch, she gets so scared. And mental anguish associated with worrying about listeria. I really appreciate your time and hopeful for response. Anyway, then she wrote also another letter. She wrote, I would pay an arm and leg for this kind of peace of mind in pregnancy. And you can see this is like I said, I never met her, I never seen her, and I know she's from Indiana, that's the state I'm from, and she keep writing. Even I received another letter 2 days ago when I'm here, and she wrote me another letter. And she asked me very specific questions about, again, some food, and she's trying to have another baby. So this would be actually second baby. So she's going through pregnancy. And she said she doesn't buy any food from anybody, she doesn't trust anybody's food, she would prepare herself. And she asked me a very specific question. Because my internet problem, time to time when I have time I want to respond, I didn't get a chance to respond to her. I'm going to respond to her with that specific question. But I just want to leave you with this thought that the listeria is a huge problem. A lot of things happens here. You hear some of the stories. Sometimes doctors may not know. They cannot link that to this. I won't say that India doesn't have that problem. It's surely there, but we are not looking for it. In the Western world, we have it. We recognize it. We take care of it because you know the problem. It is there. And so that's how we try to help them. And so this probiotic works well because of the pregnancy-related complication. We showed that in the guinea pig model. At some point in future, some point that we want to commercialize to a way that it needs to have a FDA approval because it's a GMO, right? Genetically modified organism. But you can see, at least the animal study, it can save life, right? So what we are going to weigh out, GMO versus saving life. At some point, we have to approve this. So we are very hopeful. So we are starting with the animal model first, animal application, like a livestock. If they can improve their health, we know we can have approval. And then later on in the human application, especially like listeriosis, we would be able to do it. These are some of the recent people, new people. We had two interns came from NDRI. They just started and I left. They're supposed to arrive there by April. It was so delayed. So they are, these two are very active. And then these are other students in my lab. And we have a small group, but the group is very active. Usually a lot of this work I presented, some of the students work for 5 to 6 years to complete some of the studies. And it's pretty rigorous. I want them to start one hypothesis, you complete it, before that you don't leave. So essentially when they finish, they get rewarded with the nice papers, articles, usually high impact articles. And the job they get usually, all of them are professors or some medical colleges, they work there. So they have a really bright future. So anyway, I will end it here. I know I went a little bit over time because of the length of materials I wanted to cover. But the whole idea is that I wanted to give you a flavor of type of research we do, even those probiotics we are talking about, pathogens, biofilms. And you can see this, all of those applications actually exist in this research. Right? Okay, I will end it here if you have any questions. Come on, you should have a lot of questions, right?
So my question is, let's suppose we have a biofilm in dormant stage. So sir, can the prebiotics detect it or not?
You are saying the biofilm in the
Dormant state.
And the biofilm is made by actually the probiotic. That's what you're saying?
Yes, sir.
Okay, so who is going to the immune system?
Yes, sir.
Oh yeah, yeah. So they are forming it. So what we are advantage of probiotic forming biofilm, and you know that not only in the mucus, they are coming in contact with the epithelial cells. Are you following me? So when they're in contact with the epithelial cells, they can send the signal much stronger through the epithelial cells. And the signal would be resulted to the immunomodulatory response or the junctional response, right? So those signaling cascade would work much better. Even though they are in the dormant stage, they can still do that. And that's what we are looking for, biofilm formation. So I was trying to show you the data that these engineered bacteria, they actually attach to the epithelial cells and they actually form a large colonized biofilm-like structure. And that's why we were so excited, because once you form that, attach that, form that biofilm-like structure, you don't have to feed the animal every day. We did an experiment in that paper, you can follow it, that we stopped feeding bacteria or the probiotic, then up to week and a half to 2 weeks, you can see the colonized probiotic. You see this? But if you have a regular wild type probiotic, they don't stay there. So they're in the mucus, and then eventually they go away. So that's why most of the probiotic people take it daily. Okay, but this one you don't have to take it daily because it has that property of forming biofilm. That's a good question. Yeah, if the biofilm, then some cells would be dormant, some would be active, right? And that come in contact with the epithelial cell. That's the key, epithelial cell contact.
Thank you, sir.
What do you have?
Hello. Yes, sir. Today's lecture was one of the best. Sir,
Okay,
my
great.
question is: many of the probiotics have this adhesion factors, the genes like MUB genes, sort of things, which help them to adhere. So, is there someone who is exploring that whether these genes can be used through CRISPR? Can they be used to enhance their adhesion factors in other probiotics?
That's wonderful idea. Essentially, yes, those are the approach we have to take. I don't know. I haven't read any paper along that line, but idea-wise, that makes sense. That would work very well. So some of the probiotics has the surface layer protein, right? They also make that contact. But the question is it reliable? Is it going past through the mucus to make a contact with the epithelial cell? This is the problem. The reason our system works because lab that has a strong affinity towards the HSP60. And so if there is an affinity, so if you even the bacteria on the mucus, they can pass through the mucus layer. Even they can pass through the strongly adhered mucus layer. Remember that? Loosely attached and strongly attached. Even we are showing that they go close to the epithelial cell. So even the natural probiotic, if they have these adhesion factors, a lot of times their affinity may not be well understood or strong enough to pass through the mucus layer. They need to go past the mucus layer. They need to get to the epithelial cell. That's where most of the probiotics don't work because they are way up there in the mucus layer, in the cloud, right? And they are producing the short chain fatty acids or all those molecules that diffuse through the mucus, then get to the epithelial cell. And sometimes because of the dynamic system in the gut, not all the times they are reliable. They are going to always find the positive attribute. But in our case, always I wanted to have the bacteria attached to the epithelial cell. That way you can initiate that signaling cascade and positive attributes what you saw, all those downstream effects. If you can make them attached, no matter how you do that, and that's when you can get the positive effect what we saw. Simple probiotic, we turn them into magic. You see this lactobacillus casei. Now no secret to that. That wild type didn't do protect that animal. Did you see that? Didn't do anything. Those animals are dying. Wild type has no effect. So magic, happened because you put that protein and you took that from Listeria inaqua. Now it's working very well. I know long way I answered your question. But that answer, okay. Any other questions? Come on, we learned a lot of stuff in last 10 days, right? Or I will not say 10 days, but at least 6 days, 7 days. Okay, I know a lot of times you don't want to ask in public. I know I'll talk to you in private. That's fine. Anything else? You don't want me to ask you question, right? Then you will be embarrassed. Anyway, so I hope you enjoyed it. Again, whole idea was to give you the flavor of how you can modify some of these organisms for betterment of health, whether it's gut health or disease or infectious disease. Okay, with that, I think we should take a break. Enjoy your coffee, and I'll be around. So if you want to talk, discuss more, okay, good.
Agri-Food Biotechnology Institute. That is one of the prestigious institute working in the food sector, and the institute is from Department of Biotechnology, Government of India. So Dr. Kiran Kanti, he has done his PhD from IIT Madras, and then he has joined as a postdoctoral fellow, and then he has experience of 2 years as a biochemist in Danish Innovation Institute. Later on that in 2011 he joined as a scientist in Nabi, and currently he is working as scientist E in Nabi. I am right? Scientist F in Nabi, and his area of research is gut research, gut health, and food and nutrition, and food and nutrition biotechnology. So we welcome you, Dr. Kiran, for this lecture. And just to introduce about the participants, we are having 56 students from different universities, from our university also, and from like Bundelkhand University, Manipal University, and Burdwan University. So this course is designed for like more on the microbial biofilms, and from the last 7-8 days, so they are studying this course on the biofilm, how the biofilm is happening, and what are the different advantages or disadvantages of biofilms, how we can get rid of these biofilms. And yes, so it's over to you. I hope you will like the lecture because it is more related with the food sector.
Good afternoon. Good afternoon, everyone. Is it audible? Is it audible? Hello. Good afternoon, everyone. And at the outset, I would like to thank Professor Goel and Professor Bunia for giving me this opportunity to speak about some of the research and in general about gut microbes and what are the metabolites secreted by the gut bacteria. So as he introduced, Dr. Goel, that I belong to this National Agri-Food Agri-Food and Biomanufacturing Institute, and which was previously called as National Agri-Food Biotechnology Institute. So we are the one of the frontier institutes in the country who focuses on agriculture, food and nutrition. So the agriculture division of our institute works on enhancing the plant produce or crops for nutritional traits, and food and nutrition division works on developing various kinds of functional foods using the agriculture products what we develop, as well as some other interventions like what I work on, probiotics, etc. So this is our institute is fairly new, you can say 14 years old institute compared to the other institutes in the DBT. I belong to the Department of Biotechnology Institute, Department of Biotechnology. So as I represented here, this is my research group, Healthy Gut Research Group, which focuses mainly on improving the gut health, because we believe everything lies in the gut. So if your gut is good, your health is good in a nutshell. So our endeavors are how best we can modulate this environment, right? And we use various interventions, and I basically work on developing Indian origin bacterial strains which are called probiotics, both conventional and non-conventional. And then we use underutilized crops like millets and understand how these millets enhance the nutrition security and which mitigates various lifestyle disorders. So today I will be focusing much on talking about the gut microbes and the metabolites, and some research what we do on prebiotics and probiotics to explain in a very brief manner. So when we talk about the gut microbiome, I think during this 1 week or 1.5 week sessions of this workshop, you would have learned about what are gut microbes and gut microbiomes. But to brief you again, that if you see the entire digestive tract of any animal or the human beings, it is richly populated by the various microbes, whose diversity increases as you go down to the large bowel. So the large bowel is having highest number, you can see 10 power 14 colony forming units of the bacteria. So that much of thickly populated milieu. So having said that, when they are having so much of population of the bacteria in the entire digestive tract, what are they doing, right? So what are we eating? In a way, they are also eating the same thing, and what we don't digest, they digest them, and then they produce something. So what is that something which is plays an important role? So that those things are called gut metabolites, microbial metabolites. So the entire microbiome or the composition of the microbes that are present in the gut, it can be bacteria, it can be fungi, it can be viruses, it can be parasites. Together they are called gut microbiome, but we are majorly focusing on gut bacteria, right? So, so and when you see the various genera that are distributed in the gut microbiome, so these are various kinds of bacterial species. So there is, in a normal healthy condition, there is a harmony between the good bacteria and the bad bacteria, in a sense like pathogens and commensals. So if that dynamics is breached, that leads to preponderance of the disease causing or pathogenic microbes, and that causes various kinds of diseases. So we will see like what kind of things happen. So as I said here, there are 30 plus trillion bacteria, viruses, and so on and so forth, and it has more genes. The microbes have more genes than the humans itself, right? And then we say that the various environmental factors influences the microbial composition. So during the childbirth, there are two modes of birth. One is natural delivery and the other one is cesarean or C-section delivery. So when people have analyzed the composition of the two different modes of delivery, they found that C-section has different dynamics and composition compared to the normal born. So the normally born babies have more diversified bacteria compared to the C-section, and C-section have more, like more or less like more towards the disease causing microbes compared to the normal delivery born, right? So with, as the age progresses, you can see how the numbers and types of bacteria or microbes develops, right? So in the initial stages, when the baby feeds on the mother's milk is a rich source of oligosaccharides, which are milk oligosaccharides, human milk oligosaccharides. So that itself is a big area of research, developing HMOs, human milk oligosaccharides. So the babies who are feeding on that will have higher amounts of healthy gut bacteria who are deprived of having mother's milk, right? So as the age progresses, when we introduce the solid foods, so the diversity of the bacteria changes, and again age also plays an important role. So during the progression and during the aging, there are certain species or certain genus which will disappear, and some of them are highly conserved species that remains inside. So what I mean to say, the microbiome composition is highly dynamic and changes with time, with environmental factors, with what we eat, and even that includes drugs and vegetarian, non-vegetarian, vegan, what kind of diets we have. So in the intestine, this is the intestinal border you see here. There are the mucus layer here. There is an outer mucus, middle mucus, and inner mucus layer. And then there are the bacteria or the microbes that dwell here. So anything happens to the changes in the composition of this by means of foods or any other things like diets which are rich in refined sugars or high amounts of fats. So that leads to the depletion of this layer. When this layer depletes, the composition of this bacteria changes, and the composition of this bacteria changes, pathogenic bacteria and pathogenic bacterial associated molecular patterns enters through these membranes and goes into the systemic circulation. So this is called leaky gut. I think Dr. Bunya is expert in this area of research. So that leaky gut is the root cause for most of the diseases. So in this process, what will happen, the microbes and the microbes metabolites, they enters into the circulation, goes to the various peripheral regions, as well as they interact with the immune cells that are present here and causes untoward production of inflammatory molecules. So more inflammatory molecules, they go into the circulation, they reaches various sites, and then they cause the disease. So how best we can manipulate this environment by means of diet is now an important area of research. So coming to the theme of this workshop, it is the biofilms. So it is like me playing a flute in front of the Star Wars. I don't dwell much into this. I have worked this during 2011 during my postdoc, but I have not actively pursued the area of biofilms. But little bit of information I would like to share what I know about the biofilms. So you all know by now that biofilms are aggregates of the bacteria forming a uniform layer around any kind of surfaces in their natural habitats. So they need a substrate to settle from where they colonize, and it can be monogeneous or single microbes that forms the biofilms, or it can be polymicrobial biofilms. So why these biofilms? Why microbes make the biofilms? So anyone can answer from you? No? 7 days workshop. Please answer. That is stress response mechanism. Hey, why they form? Why? Why? Why is the question? Together they want to fly, na? Together they want to sail. That makes them more stronger, right? Survive the harsh environmental conditions, right? It is a way that they mitigate the harsh environmental conditions. Imagine a single thread, you can break it easily. If you have multiple threads interwoven, it will form a big thread which is difficult to break. So same phenomena happens here. They form lumps or films. So in the lumps or the films, they stay together, and the top portion they face the heat, and the bottom portion there is all turnovers happening, new cells are forming, competition, whatever happens inside this core part of the biofilm. So as you said that they are all embedded in a exopolysaccharide matrix. They have, when you compare the free cells with the biofilm cells, their physiology is completely different. They express different kinds of genomic responses. So their transcriptomes, if you do the whole RNA sequencing and the proteome sequencing, so they might have a different kind of proteome and transcriptome responses. And all these are to mitigate the harsh environmental conditions. So we heard about much more about the biofilms that are formed by the pathogenic bacteria. Most of the pathogenic bacteria, they tend to form the biofilms. So if you take the lung is a niche for biofilm formation. If you take the intestine, there is a niche for biofilm formation. And if you have any artificial things inserted into your body like the catheters or any kind of implants, so they are the good resource or niche for biofilm formation, right? So these biofilms are almost found in all kinds of natural environments and industrial ecosystems. Common biofilms is you must have seen in the pipes, water where water runs, because there is a lots of metal deposition happens when the water flows. There is a growth of microbes that forms the layer around the inside the tunnel. So you see regularly water pipes get choked because of this. So that is one more example. And hot area of research is how the good bugs forms the biofilms, and what does that translate? What does that mean when the commensal bacteria form the biofilm? That is an important and upcoming area of research. So we did some kind of work using lactobacillus and bifidobacterium strains when I was a postdoc in Sweden, working on eradicating the Clostridium difficile biofilms using this bacteria. So there are two kinds, as I said. One is the pathogenic biofilms, which are formed by mostly by the pathogenic bacteria, and beneficial bacteria also forms the biofilms that enhances the gut health, immune modulation, and colonization resistance. So this is a bold statement, but still lots of work is being carried out in this area to understand how the beneficial biofilms helps in improving the health of the host. So this is a representation image of a mycoplasma forming the initial biofilm uh, formations here. So factors, various factors that influences the biofilm formation. I don't think I need to explain about this much. Environmental factors availability and the kind of surface that the microbes has, and the physicochemical properties of the substrate, and basically about the microbial community composition, the type of bacteria. Again, if the bacteria has more cell surface hydrophobicity, so that adheres more with the hydrophobic surfaces, and then they form the easy biofilm formation, etc. Here. So I am sorry if you are not able to see this, but this is how the biofilm formation happens. Initial planktonic cells attach to the substratum, then they form the monolayer, and then they mature, and then the biofilm cells disperses, and they again go to some other place, again find the niche, and then forms the biofilm. So like that the biofilms will be formed, and there is lot of cell to cell interactions happening here by means of enzymes, by means of RNA molecules, by means of DNA, by means of EPS, by means of proteins. They communicate with each other. So I hope you know about quorum sensing. You heard about quorum sensing molecules, AHL and AI2. So all this kind of communication happens, and then they attract various uh, other bacteria also into that. So this is what that interactions happen here. There can be gradient of pH in the biofilm from top to bottom. There is a pH changes because of the metabolic activity of the cells in the biofilm, and then there is a nutrient gradation, etc. And there is other kind of social interactions among the bacteria, and then quorum sensing is one of the important factor that allows other bacteria to come and sit into the biofilm. So this, having known about this, this creates an opportunity to disturb the quorum sensing. So there are inhibitors to disturb the quorum sensing. There are other agents that can disturb. Recently, phototherapy is one of the upcoming nanotechnology based interventions to eradicate the surface formation of the biofilms on the skin. Right? So such kind of developments have happened. So what I am more find interesting while preparing this talk, I read about the gut biofilms. So these are aggregates of microorganisms composed of host and microbial compounds. There is a cross interaction between the microbe metabolites as well as the host metabolites, and that helps in formation of the biofilms. And also there are microbes that can attach to the food particles in the mucus and epithelia of the intestine. And abnormal and deleterious biofilms in contact with the mucosal tissue, that is what it causes the human diseases and bacterial infections, etc. And beneficial biofilms, they help in colonization resistance in the sense they prevent the pathogenic bacteria to adhere into the surfaces. So they block the surfaces by means of competitive exclusion or by means of producing antimicrobial peptides or whatever it is. Right? And there is a lots of interest now on gut biofilms because thanks to the molecular techniques that are being evolved. First one is your next generation sequencing, and then the way you can look at the microbes. You can stain the microbes by means of using molecular probes, which you can tag with different colors of fluorescent dyes, and then you expose to the light, they excite and emit radiation, which in different colors depending on the wavelength. So you can literally visualize using a confocal microscopy the various kinds of microbes that are colonized in the biofilm. Initially, it wasn't possible because this fluorescence in situ hybridization and other electron microscope techniques are not so popular in the older days. So by who coined the term biofilm? Anyone knows? Sorry? I hope I gave somewhere it will come. I don't remember that. So, sorry. So this is, if you look at the gastrointestinal tract, and if you recollect the first slide what I have shown, the distribution of the microbes from the oral to the anal region. So in the stomach region, because you have high acids in the stomach, so there is less diversity of the bacteria. But still there are microbes that are present because stomach has itself some gastric mucins in it. So there are some kind of microbes like Helicobacter and Lactobacillus can colonize there, and that's why their number is less, but there are microbes. So they can adhere as patches in the stomach or as lumps and form aggregates of biofilms in the stomach. So when you go down to the colon, you can see the continuous biofilm forming cells, as well as there are some biofilms that are formed attaching to the food matrices, right. So this is imaging is not clear. So this is in the stomach. The yellow highlights or yellow color is nothing but the bacterial biofilms there, and the green ones are the glycoproteins of the mucus that is stained here. So when you go down into the intestine, you see there is a lots of yellow coloration. This is because more amount of bacteria, more biofilm formation here. So this is also represented here. This is usually a mouse gut they have taken here, and you see this in the stomach. You see less amount of biofilm, and as you go down, there is a more biofilm formation. So biofilms recently are considered, is it considered as a gut health? Indeed, if the biofilms are found with the commensal bacteria, it is it okay? Okay, I can talk like this. I can talk. I will hold and talk. No. Is it okay? Am I audible? Yeah. So, can we consider this biofilms as a marker for gut health? So, answer is yes. If the biofilms are formed with the commensal bacteria, they are more health promoting, and if they are associated with any kind of disease, they are not good. So, why they are not good? Because this is the normal condition. This is in the disease. Is it right? Disease condition. I am not able to see that. Okay. Okay. So this is the normal biofilm composition. Under normal condition, you see in the mucus there is lots of healthy bacteria that are present. So in the disease and invasion condition, biofilms enters into the inner mucosal lining, and then they are direct contact with the intestinal epithelial cells. So when they talk with the intestinal epithelial cells, so every intestinal epithelial cell is connected to the other by means of tight junction proteins, right? So these tight junctions do not allow anything go inside. So when these tight junctions are loosened, then the microbial metabolites go inside, and then they cause the disease. So commensal bacterial biofilms, they prevent, as we saw earlier slide, I told you about colonization resistance. By virtue of it, they don't allow pathogens to enter into this zone. Hence your tight junctions are protected, and hence you will have the better health. So this is the representation. You see, this is the mucus thickening. This is stained with the glycoprotein dye, which is fluorescent green color. The thickness of this defines the gut health. If this thickness is gone, the gut health is gone. So in one of the disease condition you see here, the thickness is completely gone, and the bacteria are almost interacting with the gut cells. So same here in this condition also, the biofilm is coming closer to the intestinal epithelial cells. So this causes disease condition. Right? So that is why it is important that we maintain a healthy gut microbial composition, where the thickness of this defines the health of the individual. So endogenous factors in the biofilm and host interactions. So there is a lots of crosstalk between the host molecules, host cells to the bacterial cells. So for example, the host secretes mucins, membrane vesicles, and antimicrobial peptides and immunoglobulins that can define the composition of this microbiome here. And on the contrary, these biofilms also, depending on pathogenic biofilms or commensal biofilms, they interact with the host cell. If the pathogenic biofilms are more, they interact in a negative way, they destabilizes the membranes, and they causes the inflammation, right? So that is why it is important to have a healthy microbiome composition here, and for achieving that, you need to have a good diet, balanced diet, diversified diet, which are rich in dietary fibers, omega fats, and unsaturated fatty acids, and high amount of proteins. So these are some of the biofilm images which have taken through confocal microscopy. This is not my work. This is taken from some literature. So this is Staphylococcus aureus biofilms on a polypropylene plate. They have captured it, and this is the biofilms which they have taken from the cardiac pacemakers from a patient of Staphylococcus aureus. So the point is, like, how do we eradicate the formation of these biofilms makes an important area of research. And this is some more, this is a polymicrobial biofilms. You see it is Pseudomonas aeruginosa and Staphylococcus epididymitis formation of the biofilms in the COPD patients, chronic obstructive pulmonary disease patients. There is a lots of mucus formation there, and that mucus allows the formation of biofilms of these bacteria. And once the biofilms are formed, these are the most difficult ones to eradicate. So some of the times COPD becomes life threatening because the bacteria is resistant to most of the antibiotics, because either they form the biofilms and stable there, or they would have had the antibiotic resistant variants of the pathogens. So likewise we have the dental plaques. So here they spoil your roots, and then you will have a rotten teeth. And nowadays there is a lots of research how your oral microbiome influences the gut health, because when they are in the oral cavity, they don't sit there, right? They even go into the gut. So in the gut also they causes several kinds of inflammations and infections, right? So coming to the positive attributes of the beneficial microbiomes, these are representative images of some Lactobacillus plantarum strains in a study, which provides stable colonization of the bacteria in the gut. Initially it will be like this, with time they form the biofilms like this. So some of the research suggested that the planktonic forms have one kind of biological activity. For example, if they are antimicrobial against some pathogen, the biofilm forming nature variant of the same commensal bacteria is much more rigorous in killing the pathogen. So that's why the beneficial microbial biofilms are more advantageous, because they may have a different genetic machinery, they will have a different secretome that has much more beneficial effects than the planktonic cells. So how do you disturb the quorum sensing? By means of quenching agents like biosurfactants and microbial metabolites, and some of the plant derived metabolites what I have listed here. So they do what's happening. So so these quenchers play an important role. There are natural quenchers, there are artificial quenchers, there are chemically synthetic quenchers. Not only quenchers in the form of chemical molecules, there are some physical entities also that can destroy the biofilms, that plays an important role. So this is how the molecules are prepared, these quorum sensing molecules. So if you can block this by means of genetic engineering or gene editing, so the production of these agents can be reduced, and so bacteria will not be able to attract other bacteria. That's how you eradicate the biofilms. So the probiotics, as I said, I will come to the definition of this in the later. So probiotic biofilms also follow the same pattern as we discussed earlier. But the advantages of this probiotic bacteria or any commensal bacteria is it produces various kinds of secondary metabolites. So that includes bacteriocins, that includes short chain fatty acids, that includes any other external molecules that can kill these microbial or pathogenic biofilms. So these are some of the antibacterial peptides, or roitrin and niacin, which are called bacteriocins. So they are produced by these bacteria, and then they inhibit the biofilms also, as well as biofilm form of the pathogens or planktonic form of the pathogens like E. coli, Listeria, and Staphylococcus aureus, etc. So as I also said that short chain fatty acids and biosurfactants like are also produced. They also help in eradicating the biofilms. So in the biofilms, it is they are biofilms by the commensal bacteria is a kind of biorefinery where they produce the secondary metabolites as I mentioned already, short chain fatty acids, bacteriocins, extracellular exopolysaccharides that all play an important role in improving the human health. So this is what I did work during my postdoc. At that time, there were no reports on lactobacillus and bifidobacterium strains forming the biofilms. So we have allowed to grow these bacteria in microtiter plates, and then we have stained using the crystal violet. So the more dense is the crystal violet stain, the more dense is the biofilm formation. So using that, we have seen various stressors to the, we have given various stressors to the different bacteria, and we have noted, as you some of you told, that stress as a stress response bacteria form biofilms. So when we give more bile acids, so the bacteria is tend to form more biofilms, and we have correlated this with the enhancing the cell surface hydrophobicity. So we have determined the cell surface hydrophobicity of these bacteria by various physical approaches, and then we have correlated that the bacteria which has more cell surface hydrophobicity is able to form more biofilms. So this is some of the bacterial strains which we have worked in our European Union funded project, and then using this bacteria we have developed a symbiotic composition that can eradicate the biofilms of Clostridium difficile. So this is again in presence of mucin. Some of the strains responded to the mucin because bacteria loves to be present in the mucin, and mucin is a glycoprotein which has hydrophobic patches. More hydrophobic patches, more hydrophobic interactions, and more biofilm formation we have seen in case of the lactobacillus. The same goes with the bifidobacterium strains. Some of the strains responded positively and formed more biofilms in presence of bile, while some others are not responded. And mucin had a less role except in this strain. This is a bifidobacterium lactis strain. So this is a highly aerotolerant bacterium, and which is commercially used for making bifidobacterium based formulations. So this bacterium has higher cell surface hydrophobicity and higher biofilm formation. So we have determined the production of bacteriocin that can kill Clostridium difficile. So these bacterium in presence of bile, they produce more amount of the bacteriocin and hence eradicated the Clostridium difficile. So switching the gears to the gut microbes, then biofilms and the secretory molecules. So this is the panorama of various interactions and metabolic processes happens in the gut microbes. So we all know the food basically consists of major nutrients like carbohydrates, proteins, and lipids, right? So these all molecules are getting converted into different kinds of metabolites. So whatever remain undigested by the human digestive system, when they enter into the small intestine and the large intestine, they are processed into various kinds of intermediates. So proteins are ultimately converted into amino acids. Amino acids are degraded into, depending on the type of amino acids, they are degraded into various kinds of products like pyruvates or indoles, like that. Well, if you take the lipids, they are converted into short chain fatty acids. Carbohydrates, they are degraded into short chain fatty acids. So in the gut, you have lots of products that are formed from the carbohydrates like short chain fatty acids, amino acids, and some of the amino acids are biotransformed by the gut bacteria to produce the neurotransmitters like gamma aminobutyric acid, histamine, serotonin. So you will be surprised to note that majority of the serotonin and the gamma aminobutyric acid that is produced in the system is microbially originated in the gut because they have the enzyme machinery to produce this. So and then these molecules interact with almost all organs of your body because they are produced in the body, they go into the circulation, they interact and crosstalk with all the organs and influences their bodily functions. So this is like aromatic amino acids, they are converted into tryptophan, tryptamine, and various kinds of metabolites. And recently there is a lots of research on how gut microbiome or the layers of the gut microbiome utilizes these amino acids, how they metabolizes these amino acids, and how these amino acids are converted into indoles. Indole is further coupled with so many other short chain fatty acids and form indole propionic acid, indole acetic acid, etc., which are very, very vital for the human health. So bacteriocins, you all know, they are the small peptides that can interact with the other bacteria, and then they don't allow the other bacteria to grow. These are the components, and there are various classes of these antimicrobial peptides. And even though there is lots of research is happening, but there are only one antimicrobial peptide as bacteriocin which is approved for the food industry, which is niacin, right? It is produced by Lactococcus lactis. And then you know about the exopolysaccharides, homopolysaccharides, and heteropolysaccharides that are being produced. And coming to the research what we do at NABI, so my one part of the work is to develop Indian origin strains of the probiotics. So probiotics, by now I think you all know, they are the live bacteria which we take orally in such amounts which confer the health benefit. If the bacteria confers only the health benefit in the humans, then only it is called as probiotic. Otherwise, it is not a probiotic, right? So don't get confused. Whatever you get in the market, they wrote probiotic. They write probiotic. If they are not clinically validated, they are not probiotics. It is as simple as that. So, and the prebiotics are non-digestible carbohydrates. They are the carbohydrates, but are not digested by the human digestive system, but they are digested by the bacteria that are present in the small intestine and in the large intestine mostly. So the combination of this bacteria and probiotics is called synbiotics. Syn is synergy. So 1 plus 1 should be 11, it should not be 2. So it is the rule. And the metabolites that are produced by this bacteria, they are called postbiotics. So probiotics are living, postbiotics are inanimate form of probiotics. That means they are killed probiotics or the molecules that are secreted by the probiotic bacteria. A living bacteria, if it secretes any molecule, for example neurotransmitter like GABA or any short chain fatty acid or any other molecules or exopolysaccharides, it is called the postbiotic, right? So all these play a very important role in improving the health of the human. So why we do this research is there is a lots of market potential. Indian market for probiotic bacteria is very nascent, and it has a huge potential given the size of the population of our country, right? So majority of the products which we get in Indian market are the strains which we get from either US or European or Japanese origin. How many of you have drunk Yakult? Have you seen what bacteria is there in it? Shigella? Lactobacillus casei shirota, right? So this is a strain from Japanese origin. It is from the Japanese origin. So can we develop such kind of products so that we realize the dream of our country, make in India, made in India, Swasth Bharat, right? So that's what we focus on, to develop our own strains, clinically validate them, and introduce into the market. It's not easy though. So these are the market players we have it in India, although there are some new companies, Indian origin companies also are coming up. There is recently one good bug, there is one product which has been launched in India, right? So our endeavor is also to develop such kind of product. So in one of our projects, we are working to understand what is the relation between this and this. So this is the intestine, large bowel, and this is the brain. So how your microbiome or the changes in the microbiome translates into the brain, or how the changes in the brain communicates with the gut, right? So this is the research which we started very recently, maybe 3-4 years ago we started it. And we all know that this is a bidirectional communication. Whatever molecules that are produced here, they are, some of them go here. So here they have the gut barrier and we have blood-brain barrier. Molecules to go from here to here is not easy. It is more sterile barrier than this. Only certain molecules will go. If every molecule will go, it causes damage to the brain. But still, how this communication happens? right? So this is connected to this by various means. There is an enteric nervous system, right? So the enteric nervous system is originates in the brain and innervates into the intestine. So whatever molecules being produced here, they can communicate here. So the signal goes here, influences the brain, and then the signal comes from here, and then it goes here, right? So certain kind of hormones that are produced here can crosstalk and decides which microbes to colonize here or not. Similarly, some kind of metabolites that are produced here can define your brain to function like this. So that is the bidirectional communication we are trying to understand. We have recently started, and these are all the various molecules by which that communication happens. So as I said, there are neurotransmitters. There are some molecules which can go into breach the blood-brain barrier function, and some influences various other parameters. So this is what happens, like the microbiome is disturbed, molecules leak from here, go to the blood-brain barrier, influences the brain. So this is the composition of the blood-brain barrier cells. You have astrocytes, pericytes, and some other cells here which protect the molecules from leaking into the brain. So if there is any disturbance in this, that breaches the blood-brain barrier function and causes the disease. So in our study, what we are interested is like for we are interested in a neurotransmitter gamma aminobutyric acid producing bacteria. Why we are interested here is GABA, gamma aminobutyric acid is abbreviated as GABA, and that has important function in the human metabolic processes. So GABA can influence your brain function, GABA can influence your metabolism, your digestion, your neurotransmission, etc. So pure GABA people try to take, but that is not advisable, because anything you take in a pure form in more amounts on a longer periods, it will have its own demerits, right? Anything in excess is not good. Even good becomes bad when you take more, right? So our endeavor would be, if this GABA goes and colonizes in the gut, whether it will produce GABA, and that GABA influences your brain function. So whether the bacteria is communicating, or the GABA that is being produced by the bacteria that communicates with the brain, that is what we are trying to understand. So in the first part, we have isolated the bacteria, and we have identified by 16S based identification as well as whole genome sequencing as Lactobacillus brevis, and it has the machinery to produce gamma aminobutyric acid from the glutamate. So glutamate, upon decarboxylation, it goes into and it becomes GABA, and that GABA plays an important role. So it can be, glutamate can be taken inside, it can be converted into glutamate, or glutamine can be converted into glutamate, can be converted into GABA. So this bacteria has both the enzymes that can convert glutamine to glutamate to GABA, right? And then this work is published recently, and whole genome sequencing also we have done, and we have done the safety study of this bacterium by certain safety parameters, toxicology and safety parameters, subchronic, chronic, and subacute dosing in animal models in both sexes. And interestingly, we have done an experiment. How many of you are aware of epilepsy, seizures, fits? In a common language, it is called fits, right? There is a movement of unwanted movement of your limbs and legs, and everything happens, right? So that is because in conditions of epilepsy, there is more excitatory stimulus nerve activity than the inhibitory nerve activity. So that inhibitory nerve stimulus is by gamma aminobutyric acid, or GABA, which is low in these people who are suffering from the epilepsy. So taking that, we thought that whether that disease can be prevented when we give this GABA producing bacterium. So we did not give any GABA to this bacterium, we give this bacterium to the animals. So this is a preventive study what we have used. Like first we give the animal, we feed the animal for certain time points with this bacteria, and then we give an agent which causes the seizures. So this agent is nothing but pentylene tetrazole. It causes seizures because it kills the GABA energetic function of the nerves. So animal becomes more excitatory, then it has certain kind of movements which I have listed here. So during various interventions, before that we give this bacterium, and during the injections of this agent which causes seizure, we also give this same bacterium. We continue till the experiment. So the behavior of the animal is given as the resin score. More the resin score is more the seizure. So this PTC is this one blue color, which causes the disease, pentylene tetrazool. The more the score, the animal has more movements of the leg, hind limbs, forelimbs, entire body. I will show an interesting video about it. And this is the standard drug which is used to treat the epilepsy, valproic acid. And this is the Lactobacillus brevis, which produces the GABA. And this is the strain which produces GABA but in less amount. So there is a clear difference between the two strains, and the disease and the positive control, you see there is a huge difference, right? And we have analyzed various neuroinflammatory parameters in the brain. So you see that inflammation is very high here, and the inflammation is reduced upon feeding this bacteria, right? So we also saw the dendritic spine densities in the certain regions in the brain. More the spine density regions, more is the protective mechanism. So in this case, you see these are the protrusions, you see these are the spine density in the healthy animal. In this you see there is a decreased spine density, right? And this is the spine density when you give with this bacterium in different regions. You see there is a significant improvement in the spine density. So this is what we have quantified. We have measured the different regions under the microscope, the density of these spines in the neuron, and then we have given the quantification. And this is a study which shows the colonization of this bacteria. So we have labeled this bacteria with a fluorescent dye. We have tagged it, and we have followed until 8 hours after you give the first dose of the bacterium. We have followed how the bacterium colonizes by using a special visualization machine. So you see here, when you give the dose, this is the dye, normal dye. In 8 hours of time it reaches here, and this is the bacteria labeled with the dye. This is the initial phase in the oral region, and then you see that it is going. This is the stomach region, and then it is in the intestine. After 8 hours also, you can see here the bacteria is being colonized. So that means this bacteria has the ability to colonize into the mouse gut. Sorry, this is the rat gut, but we do not know post 8 hours what is the fate of this bacterium, because this dye is active only for 8 hours. Beyond that, the dye automatically destroyed. So we are following it up with deriving the strain specific primers, and see through fluorescent in situ hybridization upon different time points. After we first feeding, you stop the feeding, and how long it will colonize in the gut, right? Although it is not essential that bacteria do colonize there, even if it transiently colonizes, if it is doing the job, that is enough, right? And then we have also studied 3 different doses, and we understand by our experiment that the middle dose, which is 2 into 10 power 10 CFU of this bacterium, is much better in protecting the animals. So this is that interesting video what I talked upon. So can I keep this? I want to play the video. Yeah. You can see here. So this is the control, this is the disease, this is given with the lactobacillus, this is a symbiotic product we prepared, and this is the drug control. You see the amount of protection that lactobacillus is exerting. So by now we know that this bacteria is able to reduce the disease, but why? But what mechanism, we do not know. So we have to do some kind of follow-up studies. We have to cut the vagus nerve, which is the main communication between the gut and the brain. That will give some idea. And then we have to use germ-free mice. Germ-free mice don't have any any microbes in their gut. But if you colonize with this bacterium, so you know that this bacterium, what it is doing to that. So such kind of studies, follow-up studies we are planning now. And this is another study which recently got published in Journal of Applied Microbiology. So we have developed a symbiotic gland which comprises of 4 strains of the bacteria and a prebiotic isomalto oligosaccharide. And this we have fed the animal with high fat and high sucrose diet. You see that this rat has become bulky, and when this symbiotic supplement is given to this animal for 12 weeks, so that is, it is preventing the weight gain of the animal. So how it is doing? Partly we suggest that it is preventing the gut dysbiosis. We have analyzed the expression of tight junction proteins in the intestine, as well as we have seen the western blots. We have done the gut barrier function analysis by feeding with dextran, FITC dextran, and we saw how much it has circulated into the, transferred into the serum. So such kind of experiments gave us a clue that symbiotic supplement protects the gut barrier function. So leaky gut is reduced. At the same time, it modifies the gut milieu, and through production of short chain fatty acids, reducing inflammation, etc., etc. So overall, my group works on various aspects. One is millets and bioactives, how it helps in the gut barrier function, how manipulates the gut bacteria, and symbiotic blend, how it communicates with your adipose tissue. And recently we started working on this GABA and GABA producing bacteria, understand gut brain function. Right? So with this, I would like to stop my talk, thanking the organizers of this workshop, Dr. Gunjan, Dr. Bunia, for giving this opportunity. I would like to thank various funding sources. These are various funding extramural grants we receive from various funding agencies. I would like to thank my organization for giving me this opportunity, infrastructure and core grants every year for conducting this research. And these are my collaborators, Dr. Mayanur Ismail, external collaborator, Professor Kanwalji Chopra from Pharmacology Department of Guwahati University. And this is my boss, postdoctoral boss, Professor Torkel, who taught me the life in the mucin. So every time we used to discuss, he was like, we have to work on mucin production, because how bacteria interacts with the mucin. And we did so many kind of experiments using biophysical methods as well. So such a nice, wonderful, passionate researcher. He is an expert on Helicobacter pylori. So his entire life is spent on understanding how Helicobacter pylori dwells, causes gastric cancer, and he is the one who discovered, other than Helicobacter pylori, the other species of Helicobacter. And these are the PhD students whose work I have presented. Tushar is working on this GABA producing bacteria, how gut and brain are communicated, and Anita is working on developing the symbiotic, so she is about to submit her thesis. So with this, I would like to thank you all for your patient listening. If you have any queries, you are most welcome to ask.
Good afternoon, sir. Sir, my question is, as you informed us that gut microbiota is very crucial, so then why do some germ-free animals still die?
Germ-free animals,
Still die.
they die because it's a natural process, right? See, germ-free animals had a life different from animals which are colonized with this bacteria. So they have a compromised lifestyle. Even the knockouts also. Knockout animals means you knock out some genes from the animal. They also have a compromised life. Although they live for some time.
And sir, in some cases, as you know that identical twins, they have same genetic composition and same eating habits, that their diet is same, but still why they have different microbiomes?
I don't know. No answer. You tell.
So I don't know actually.
Maybe sir can answer it.
Yes, sir. The same location, then why do they still have different microbiomes? Yes, sir. Same diet, same genetic composition, everything same. No, sir, I'm just supposing this. Yes.
See, See, even they are genetically similar, some kind of genes may be active, some kind of genes may not be functional. So there are several factors. So in some of the experiments on the lab animals, you have a cage, right, in a shell. Upper shelf has different microbiome than the lower shelf. So they are in the same atmosphere, right? But there are studies which says that.
Thank you, sir. That was I was asking why it is so.
Environment, environment plays an important role. Even though we know the genetic makeup, we do not know what kind of metabolomes we have. It also differs. Right? So metabolites itself can influence the microbiome composition. For example, your hormones, steroid hormones, plays an important role in regulating the microbiome. There are so many. See, microbiome science is not easy, but it is a good question.
Thank you, sir.
Good afternoon, sir. My question is can you highlight the need of bioengineered probiotics in the India for the Indian population or in the Indian market?
See, to understand the mechanisms, we need a bioengineered probiotic. But Indian context is a different niche actually. In India, even normal probiotics, you say people ask, why do you want probiotics? To be honest with you, nobody is interested. They say we are eating so much of curd, we are eating so much of fermented food. But if you go by the definition, you have to eat in sufficient quantity. You say daily I am eating curd, but does it have a microbe which do the function you want, and how much amount you have? So this depends. So engineered microbes is not approved in the Indian context as far as my knowledge goes, because we are not allowing transgenic plants itself, crops itself. So microbes, you forget about it. But for understanding, for being prepared, like COVID kind of emergency, you might need a microbe which is engineered. So people try to do their ready things to do this. I know. Please, sir. Please.
Only we are adding probiotic organism to fermented products, and that's really popular. When you go to grocery store in US, we try to buy probiotic yogurt or curd, and those actually, if you look at it, Chobani is one of the company. They add extra microorganisms to the yogurt after the fermentation. Do you see this one? So once it is fermented, the yogurt is made, and then they are going to supplement with, say, Lactobacillus rhamnosus or Lactobacillus kgi or all of those organisms, or also bifidobacterium. Now they are selling as called probiotic yogurt. Before that, if they don't have this probiotic organism, they don't claim as a probiotic yogurt. And you pay $2 or $3 more for that probiotic yogurt. And if you take that, then you get the probiotic. Otherwise, there is no proven probiotic effect. That may be argument. We have a misperception. We are calling fermented products as a probiotic. Generally, they are, but you have to think about what organism they are using to ferment it. One of the Korean product is the kimchi. Kimchi is real popular, and they're finding out the microbiome, the probiotic action is through the roof. It's almost you can eat every day, the kimchi. I don't know whether you have tasted or not. It's a fermented cabbage. Have anybody tasted kimchi? Some of you tasted. And they make it very spicy also, different flavor. But Korean, they won't survive without kimchi. Almost like every meal they will eat, starting with breakfast, lunch, dinner. Koreans, yeah. So that's one of those, they claim really good probiotic organisms they have. They have proven some of those.
Even in case of fermented foods, like due to the action of these bacteria, you generate so many active peptides there. If you take example of yogurt or any curd, your casein is converted into bioactive peptides having this angiotensin converting enzyme activity, then DPP4 inhibition, so that controls your blood pressure and diabetes. So it is not the bacteria is doing directly, it might be doing through the production of bioactive peptides, which is a misleading to call yogurt as a probiotic.
That's right. That's a good point. Any other questions?
Good afternoon, sir. My question is why the infant gut microbiota composition differ according to the mode of delivery?
Mode of delivery. C-section means you are just pulling out the baby, right? And in a natural delivery, the baby goes out through the vagina. It has its own microbiome there. So in the process, the baby acquires some amount of microbiome from the mother, which are born through the C-section. Through the natural delivery. That is the only people gave the explanation. Caesarean has a different microbiome compared to the natural. Yes. That's what I told now. See, in caesarean you will cut here and you will pull out the baby, while the baby has to transit through the vagina, which has lots of microbiome. There is a lot of research happening on the vaginal microbiome, how it can be manipulated, because most of the time they have this urinary infection. So because there is a compromisation in the vaginal microbiome composition. So baby born through that is expected that it will take some microbes from the vagina. That's right.
What's the mechanism?
It's through oral route or through any surface interaction. Even they say earlier, meconium is when it is inside the stomach. Now it is a questionable. Earlier people used to think that it is sterile, but now they are saying that meconium is one source.
But it is a natural process. I think when the baby is coming through the vaginal tract, and the tract is colonized by a lot of baby microbes, that's what keeping the system open. And so as the baby comes through, remember, oral opening, right? And the baby absorbs the skin, all of those organisms are there, right? And when the C-section is that we don't have that option of having those microbes from the duction.
Yes, sir.
C-section organisms come from the nerves.
Through your handling environment.
So, sir, like, is there any difference between like preterm babies and like which are fully mature? Like some babies are delivered at
There are
7
studies
or
which says
8 months. Yeah?
there are different.
There are differences.
It is simple to predict also. Preterm is before the
Like
entire
less than
gestation
37
period.
weeks.
Some babies are born even in 7 months. 6 months. I have seen a case where babies were born after 5 months. They have to put in the insulators or
like
so they will be fed with artificial milk from outside. It has a different microbiome.
okay, sir. Thank you.
Okay, so at the end of this session, I would like to thank Dr. Kanti for his talk. Although he has done a wonderful job to combine the biofilm, because I requested him to say something about the biofilm. Although it was long back he did this work, but he has wonderfully combined the biofilm and the gut microbiome and his work on the millets and the society fiber, prebiotic substrates. So I request Professor Bhunia just to felicitate our guest.
reshuffle the schedule. So let's talk about that antibiotic resistance. The role of biofilms in antibiotic resistance. Before I start that, I think the C-section and natural birth, this is something in a very interesting area. We looked at a lot of papers, and there are a lot of research articles. Um, there are, you can look at and read, and you will find a lot of knowledge. And this is something extremely important in terms of getting started with good microbiome, right, in early in life. And then as you progress, your microbiome changes, right? And then the microbiome becomes very unstable early in life. As you in the middle, then it stabilizes. Then when you are getting old or older, then the microbiome again destabilizes. Okay, so there are very interesting research in this area. Anyway, so we're going to talk about antibiotic resistance, and I know you are. I think a lot of you asked many questions about antibiotic resistance contributed by biofilms, right? And again, you remember the layers we talked about, how they are organized, why antibiotics are not reaching to the microbe in the biofilm, what are the factors, and what are the natural process of microbes being resistant, right? So in general, I believe all of you have very good idea about antibiotic resistance, right? If you have taken any microbiology course and they talk about antibiotic resistance, there are a set of mechanistic aspects, right? But we are going to study from the biofilm standpoint, right? How biofilm contribute to the antibiotic resistance. So obviously, I'll try to tell you in a way it's very clear to you, and I don't think I have many slides, but enough to convey what is there and how the biofilm contributes to the resistance. Okay, so antibiotic resistance or antimicrobial resistance, they call it AMR. It's well recognized, right? We recognize the problem associated with AMR, and this is prediction. Prediction is that there, the mortality rate, they're expecting 10 million per year by 2050. It's obvious, right? If you suffer from a bacterial disease and only treatment option is antibiotics, and if the organism do not respond to the antibiotics, obviously it becomes fatal or fatality for the patient, right? So that's the prediction. So there is no set antibiotics you could use for treatment, right? And so if that's the case, you're still trying to be entertaining or what? No? Okay. So we need to find an option, right? So obviously that prediction is serious, right? And it can happen unless we do something. So this is always happens. If you see future numbers, that's to scare you. That's the whole idea. It may happen, it may not happen. But the whole idea is that if I bring that to you, knowing that this problem may come someday, then as a scientist, you will be better prepared. Can we develop alternative strategy? So few of those things we are going to talk about. Multi-drug resistant bacteria is a problem. We know that if an organism resistant to multiple antimicrobials, so that's called MDR, multi-drug resistant. And Salmonella is one of them. Sometimes they could be resistant to 8 antimicrobials, okay? And also some of the other ones are extended spectrum beta-lactamase group, which are Acinetobacter, Klebsiella, Mycobacterium could be also, and then Staph aureus definitely one of those organisms are resistant to antibiotics. WHO reports 490,000 cases of multi-drug resistant TB, and if TB, that was in 2016, and think about that, tuberculosis, if it started to flare up and the drugs are not working against it, it's a serious consequence. We know that it's one of the deadliest diseases. I know this slide may not be easy to read. What you are seeing actually, the different types of organisms on the left and number of cases we see in percentage, okay? And so the Staphylococcus aureus at the bottom, and Actinomycetes, or Actinobacillus baumannii. That one, Actinobacter. That's in the high. So we're talking about the resistance or this is very hard to read some of those things. Treatability, mortality, healthcare burden or healthcare cost. This is what they estimated. That's essentially what this slide says. This was published in Lancet, so you can follow this, and saying that this antibiotic resistance problem is a serious concern to the humanity. Again, as I said before, that you already know the different mechanistic aspect, how bacteria or microbes become resistant, right? So this is an overview to summarize that if the antibiotic comes in, there are different mechanisms, right? So one is called diffusion barrier. That means the microbes may have outer components, whether it's a mucus layer or the outer membrane layer, or they are producing capsule, any of those molecules that creates a barrier, right? The drug comes in, but there is no way of getting in. That's called diffusion barrier. So that's one of them. Then efflux is pretty common, or one of the common mechanisms. So that means they can take the drug, but then spit it out. That's the efflux pump. Then also target modification. So if it is going to bind to a certain target to inactivate the microorganism, and then the microbes as a defense strategy, it can modify that. So for example, protein synthesis or DNA replication. Any of those targets being targeted by the antibiotics, the microorganism can modify it. And so that's why it's called target modification. Metabolic bypass. So they can bypass the metabolic process, so therefore they don't have to utilize the drug. Or drug can alter the metabolic process as well. They can also enzyme, antibiotic altering enzyme. Enzyme can inactivate some of those drugs. So therefore the bacteria is making that enzyme. And so that enzyme can break it down, right? One of the example is the beta-lactamase. We will talk another slide. But say for example, if it breaks down the penicillin, so penicillin is no longer going to be effective against cell wall synthesis. Okay. Then antibiotic degrading enzymes. So this is altering enzyme, and this is antibiotic degrading. That's what exactly what I mentioned. So these are the standard strategy. So this one, again, I don't know whether you'll be able to read it from a distance, but once you get the slide, you get a very clear idea. And I thought this slide is very distinctly descriptive. So for example, this is saying the structure, right? Outer membrane, cell wall, cytoplasm, and the lipid bilayer. And this is DNA replication. So some of the drugs you take, say quinolone, right? They affect the DNA replication, okay? And the main function is the topoisomerase enzyme, okay, which helps in the DNA replication. And so this is one of the targets. So the rifamycin, this works on DNA-dependent RNA polymerase, okay? And this is also the protein synthesis inhibitors. These are the group of antibiotics. Then this is also the folic acid metabolism inhibitor, like sulfonamides, trimethoprim. These are the drugs. They work by modifying the folic acid synthesis. If it inhibits folic acid synthesis, and bacteria or microbes need the folic acid, they don't have that. So essentially, it's kind of indirectly killing the microorganism. Okay, and cell wall synthesis inhibitor, penicillin, for example, right? Vancomycin. There are a lot of antibiotics that block the cell wall synthesis of the bacteria. So these are the group of antibiotics that are responsible for inhibiting cell wall synthesis. So when you get this slide, read it closely. This is really, this paper, I liked it because the way it explains you how different mode of action for each of the antibiotics. Okay? All right, so this is background. These are again kind of like a reinforcing the thought I gave you. So this is saying that classes of enzymes involved in antimicrobial drug resistance. So these are the different classes of enzymes that modify the microbial or antimicrobial drugs or antibiotics, right? So let's look at the penicillin, for example. This is the beta-lactamase activity or beta-lactam, what it does, I told you already that it acts on the peptide cross bridge, which holding N-acetylmuramic acid, N-acetylglucosamine structure, and then the bridge formed by the peptide, and the beta-lactam cleaves that. If they cleave that peptide bridge, therefore the cell wall structure become unstable and that fall apart, and that's how bacteria die. Okay, so this is example being used how this penicillin works. Enzyme modifying action, again that hydrolases or transferases or redox enzymes, these are modifying the enzyme, right? So this modifying enzymes made by the bacteria, and therefore the drug doesn't work. Okay, try to again simplify the mechanistic aspect how the drug resistance evolved. So the antibiotic resistance kind of briefly mentioned the mechanistic aspect of different antibiotics. So here, why we are getting this antibiotic resistance? Why there is a problem? And I think all of you have some idea, right? I mean, I listed 3. So probably you can come up with the reasons why we are facing this problem, right? So let's talk about the general one. Antibiotic use in animals. You knew that they use as a growth promoter. You heard about it? In livestock animals they will feed antibiotics. Why? Why they will add antibiotic in the feed? You heard about it? Whether it's a poultry feed or swine feed or cattle feed, they will add certain amount of antibiotics. Increase their growth, right? Increase their weight. Why? Because what they are trying to do is that they are trying to control the microbial population in a way so that the metabolic byproducts, which are the protein built up, animal gain weight. It's not therapeutic dose, it's more subclinical dose. And sometimes also animals suffer from some mild infection, right? And if you give them antibiotics, you can control that infection. What's the problem with infection? Because the immune system is working hard, right? What about the cost of active immune system? Think about why. Always ask that question, why? We add antibiotics to control subclinical infection, right? We all agree. The reason is, if you have a subclinical infection, your immune system has to work hard, right? And every aspect is going to cost you, right? What is going to cost you? More energy, right? So the food you are eating, you are actually directing that energy towards the immune cells so that they can function better. What happens to the most important aspect of the livestock? Growth, right? That's the conversion, or the milk production, or the egg production. Are you getting it? Why we are using as a growth promoter? Because the animal, if the broiler industry, if you start feeding them feed, and if they don't gain weight 2 to 3 kg or 4 kg in certain time, that means it's a loss, right? So the feed to meat conversion, okay? But if there is a subclinical infection, certain microbes are engaged, your immune system is highly active, and the energy is going towards those immune cells to work better. So you see this balance? Therefore, your energy is not helping the growth of this animal. So that means economically, you are not gaining money because all those resources going towards fighting the disease. Like when you become sick, you lose weight, right? Natural. And no matter how much you eat, you still don't gain weight. But when your natural infection goes away, then you gradually come back. Because all the food you eat now is converting that into muscle, the meat, right? Or lipid or fat accumulation. Everybody clear with that? So that's why they practice using sub-therapeutic level antibiotics. But what's the problem with the sub-therapeutic level? Everybody understand the therapeutic level? Therapeutic? What do you mean by therapeutic level of antibiotics? So if you go to see a doctor, right, and they will prescribe you antibiotic, and they will tell you that you have to take this antibiotic for 10 days. They will give you certain numbers, right? And they will tell you the dose. They say 500 milligram of that drug or that tablet or capsule you have to take it, right? And that 500, once you take it, your body or blood circulation maintains certain dose, okay? And that dosing is at the level that can kill the pathogen. Are everybody clear? So you need to maintain that dose. So that's called therapeutic dose. Therapy more for treatment. That's a treatment dose, right? So at that treatment dose, if you follow that 10 days of treatment, it's supposed to take care of all the pathogens from your body. I got it? So that's how it cures it. So you have to maintain therapeutic dose. So the whole idea to eliminate all the pathogens. What if you take less than therapeutic dose? That means subtherapeutic. Are you getting it? Subtherapeutic, is it going to kill all the pathogens you have if you are suffering from a disease? No, right? So those animals, they are not suffering from a full-blown disease. This is like a subclinical infection. That means there may be mild infection, E. coli or Salmonella growing, and therefore they try to use this antibiotic randomly so that these organisms do not have a chance to flare up. Everybody clear? What is the problem with the subtherapeutic dose from the resistance standpoint? You are not killing them totally, but you are helping those microbes to become resistant. Are you getting it? So if you keep on using these antibiotics in animal feed, we are creating microbial population. They are becoming resistant because we are not killing them, but we are making them more resistant or tolerant. Are you getting it? So that's the problem with the feed. So now the regulation, at least some European countries, I think they banned a lot of antibiotics. And then US, they haven't banned it yet, but their regulation, they are asking those animal feed, you should use certain antibiotics which are used primarily for animal diseases, not for human diseases. So now we are talking about totally different thing. The antibiotics used for human infections, you should not use in the animal feed. Okay? So they are trying to define some of the antibiotics they use only for animals, they want to use them in those subtherapeutic as a feed additives. But then again, that's also not good, because even though you are using those veterinary used drugs, DFD, I think that's the veterinary VFD, veterinary feed directive or drug, I think that's the terminology they use. So the whole idea being is that if the microbes are resistant to one antibiotic, what did I talk? I spent a lot of time talking about central regulatory element. Remember, you have to connect them now. Yeah, so if the central regulatory element become resistant, I use that example, and then all the genes underneath, they can show cross protection or resistance to other antibiotics. And so what's happening also, any microorganisms exposed to one type of antibiotics, they can show resistance to other antibiotics. Okay, that's also another problem with this antibiotic resistance. So indiscriminate use by humans, we all know that, right? You go to pharmacy probably in India or some of those countries, it's not regulated. You go to pharmacy and you ask for antibiotics, they will give you, right? But in US, you cannot buy any antibiotics without prescription from a doctor. You cannot buy any. And then other directive, make sure you follow the dose. A lot of times people, what they do, they feel good after 3 days and stop taking that. Don't ever do that for antibiotics. Or even if you are someone taking it, make sure they complete the course. It is so important, because otherwise you are generating persister cells, right? We talked about resistant cells, and then your microorganisms, they are not completely cured, they can come back as soon as you stop the antibiotic. So that's why the completion of the antibiotic dosing is very important, right? So the indiscriminate use is this. You are suddenly ill and you just go and get some antibiotics, right? And then you stop. But you don't usually take antibiotics. That's my advice. Try to avoid it. And that's the practice we have to do. And the other thing is that a lot of people or doctors prescribe antibiotic even though you are not suffering from bacterial diseases. That happens. Sometimes you are just having flu, right? some kind of low fever or high fever, and then suddenly first thing they will do, give you antibiotics. But we know antibiotics work against only bacteria. They are not going to work against viruses. Are you getting it? But sometimes I remember when I was doing veterinary practice, if the animal come to my clinic or I'm seeing a patient, I would add antibiotics. There is a reason for that also sometimes. What happens is that if there is a viral infection, virus infection also can suppress immune system. Okay? And therefore the bacterial infection become dominant. And it happens. So that time antibiotic can control the bacteria but not the viruses. So that's a secondary effect. That happens. There is a reason for it sometimes. But naturally, if you are suffering from, if you know you are suffering from some kind of flu, okay, or some gastric ailment, which you know it's called self-limiting, means after 2 days, 3 days of rest, you can recover. Okay. So you have to make the decision. Also, the doctor has to make the decision if you are going to see a doctor. Okay. If you don't do that, then randomly we just prescribe antibiotics. Definitely, we are going to see this bad effect. Okay. That happens. Other thing, have you ever thought about that? What do you do? The leftover antibiotics. How do you dispose it? Where does it go? Or all the drugs you take, the drug you are not consuming, right? You are throwing it away somewhere, right? Where do they go in the environment? Have you ever thought about it? Any kind of drugs, any kind of medicine. When I say drug means I'm talking about medicine, right? What do you do? Where does it go? Think about this. If you are draining them in your sewage system, the water in your sink or toilet or drain, look at this, where they end up. They become in the water, in the soil, right? If they are in the soil, in the water, if those drugs are still there, right? Who they are going to interact? What we have in the soil? What we have the plant? Microbes everywhere, right? So what we are doing now? Think about it. A lot of those pathogens we deal with, they come from soil, water, plant. They already have antibiotic exposure because you did it, or the company who are making it, they're draining stuff in the environment. So think about the cycle. So don't blame it for one group for antibiotic resistance. I think we all contributing to the antibiotic resistance. See the cycle now? You should be able to connect it. So throwing it in the soil, throwing it in the water doesn't mean that you're getting rid of it. Yes, it's not in your body, but it's outside. But those microbes, they are becoming resistant now. And someday they are going to infect you back through plants or food system. So that's one of the issue also with the antibiotic resistance. Then the last one, transgenic crops. What are transgenic crops? Genetically modified, right? We are creating hybrid varieties. Okay, so this one suddenly dawned on me that yeah, how that transgenic plant can be a source of antibiotic resistance. Transgenic plant. So if you're taking corn, for example, or soybean, or pumpkin, or squash, a lot of those products, they may have gene, right? And when you introduce gene, usually they are attached to certain antibiotics. Antibiotics we use as a marker. Okay? So transgenic crops, think about this. GE crops, we call it. Over 100 crops are GE. Corn, apple, potato, soybeans, cotton, sugar, beets, canola. A lot of those are actually have a gene. And a lot of times we have to put this antibiotic marker. Think about it. You never thought about that plant materials could be a source of antibiotic, and they can also transmit. So how? Soil, water, the microbial community, then food, human, insect, animal. So all of us in this whole ecosystem where we live, everybody can transfer this antibiotic resistance genes. Okay, and so that's the point you have to always remember. Now let's look at the spread of antibiotic resistance gene. How do they spread? Okay, we are going to talk about biofilm in relation to biofilm, right? We already gone through this. So if you look at this slide again, maybe BG, I don't know whether you'll be able to read it. So what we are looking at, how these different factors contribute to the antibiotic resistance, right? So you have to now think about the biofilm structure, right? So they are in a closed community. If I have to summarize it, so if the antibiotics coming in from the top, right, we had polysaccharide, and so that's going to prevent the penetration of antibiotic, right? Or they are going to absorb the antibiotic. So the antibiotics may not reach to the microbes that are in the different layer. So the top layer would be always protected by EPS or eDNA, or the oxygen concentration is low. And other aspect also, these microbes are metabolically inactive, kind of like dormant, right? And the antibiotic mechanism, the antibiotic has to be actively taken inside. Which cells are going to take it inside? The growing cell, right? Dividing cell. For example, penicillin, right? Is penicillin going to work against if the cell is sitting there in one location, not dividing? Is it going to work? I know you are tired. Let's think about it. So the bacteria, the penicillin targets cell wall, right? So now a cell not dividing, sitting there, is it going to be affected by penicillin? No, right? Resounding no, that's good. That's strong no, right? No. Because what happens, the penicillin is going to absorb only when the cells are dividing, right? Already the one already divided and already static sitting there, penicillin is not going to get there and going to break it, very rarely. But only the uptake would increase as the cell divides. You see this antibiotic is there, and that's going to stop the cell division, because cell wall become very porous. So a lot of those antibiotics work when the cells are metabolically active. They can take up stuff, and now it's going to kill it. But if you are sitting there dormant, yeah, no effect almost, because your system is physiological system is kind of like a shut down from everything. You are not dividing. If you are not dividing, that means what? Your DNA is not replicating, right? The topoisomerase enzyme is not going to work. So the quinolone family of antibiotics is not going to work. So all of those things works on actively growing healthy cells. Will be clear on this? So that's one reason, or primary reasons, the cells in this biofilm environment, which are stressed, which are dormant, which are metabolically inactive or slow, they don't have much response. And that's one of the reasons we get strong antibiotic resistance. Everybody clear? So these are the many factors you will see. So the degradation would be affected, efflux would be affected, acquisition of resistance determinants, the gene expression, and some of those things. Okay? All right, so that's one way to look at it. Then when they are in the community, for example, they are early in the maturation process, right? The microbes are still active, right? And are they going to show resistance? So let's look at some of the other factors. So biofilm forming bacteria are embedded in a matrix, we all know, and they are highly tolerant to antibiotics, UV light, chemical biocides, host immune response. These are some of those things we already talked about, right? So let's look at, I think these are the different types of cells, right? So the quorum sensing taking place, communication. Then we can have viable but non-culturable cells. What are those cells? They are metabolically inactive, right? So they are not going to take up a lot of stuff. They are sitting there. Persister cells, we talked about the cells which already is kind of like a dormant cell, right? Very close to like B, B, and C cells. Resistant cells, yeah, we know they are naturally resistant to antibiotics, right? Probably they are not going to respond to antibiotics. They are not going to get killed. And then also tolerant cells. We had a discussion between resistant versus tolerant. Resistant is they can grow in presence of antibiotic, right? Tolerant cells, they may grow up to a certain level, then they will stop. That means you increase the dosing, they will gradually slow, and they stop. That's the tolerant. Everybody clear? The resistant organism in that environment, they will continue to multiply because they don't care. Everybody clear? And so those populations you have in this mixture. Okay. So the mechanism of antibiotic resistance in biofilms. Let's examine resistance at the biofilm surface. We recognize that we got the matrix, pore diffusion, slow penetration of drugs through this complex matrix. That's recognized, right? Resistance within the biofilm microenvironment. That's the natural phenotype. Some of them are, if you have a multi-drug resistant salmonella or actinobacillus or Klebsiella, they are all in this biofilm complex. They are naturally resistant to many antibiotics. So obviously they are going to survive well. Resistance of bacterial persister cells. Again, keep on talking about the persister, because the toxin antitoxin effect, they're making them survive in that environment, still kind of dormant, but they are not antibiotic resistant. Okay, they've gone through that phase where they don't care about antibiotics, they don't respond to antibiotics. But as soon as you remove that pressure, they revive back and they can respond to the antibiotic. So these are again components of the matrix that can hinder the absorption of antibiotics in the biofilm. We already talked about polysaccharide, exopolysaccharide, eDNA, lipid, protein, all of this is going to prevent penetration. Resistance phenotype we already talked about, and then within the population of biofilm, there is a heterogeneity. That means again same thing we are repeating. Some are sensitive, some are resistant, so all different stage of in their life they are in this configuration. So their response would be different. So if you are saying that some of the cells getting killed on the top, the one at the bottom they are still surviving, there is no antibiotic, and then they can flare up with the substance, right? They can grow within that biofilm. So their susceptibility varies depending on what stage of their life cycle within the biofilm. Other thing also we talked about a little bit, some of the speakers also mentioned horizontal gene transfer. Because we are all in this community, always genetic exchange can happen. And that do happen. I'm just going to example some of the ways that happens. So 3 different ways gene transfer happens, right? If you are taking molecular biology, we know that. We know that the transformation, right? Genetic material can get into by penetration. You can weaken the cell wall, then it can be taken in, right? Then transduction, usually by infection, right? Viral infection. And generally bacteriophages, if you encode it with the gene of interest, you can introduce that, right? That's one efficient. And other is the conjugation. We know that fimbriae or pili, they will exchange their genetic material. So these are the standard practice, it happens. These are actually just a graphic they are showing what are the different ways these bacterial organisms, again same thing what I just mentioned, okay? But have you heard about microvesicles? Okay, again go back to the cell structure, the gram negative, gram positive, think about their outer membrane. So what happens, these vesicles, these are part of the structure, okay? Some of those bacteria, they will release small vesicles, and those vesicles are packed with nuclear material. And so other bacteria in the vicinity, they can accept it. And this microvesicle strategy is not old, but a lot of times people don't pay attention to. Is that contributing to pathogenesis, also antibiotic resistance? So what happens is that these are different names called BMVs, bacterial membrane vesicle, outer membrane vesicles, OMVs, membrane vesicles, also MV. So these are the different names we use it. So essentially, it's like take the cell, right? It's round or long, then outer membrane. So think about outer membrane came off the bacteria, and since it's a lipid bilayer, it can form also like a balloon-like structure. And when it is out from the dead cell, it can carry some gene. It carries a lot of stuff, a lot of toxins, also the genetic material that will be released by these cells. And so OMV transfer beta-lactam resistance plasmid to Klebsiella pneumonia. So there is evidence this happened in Klebsiella. OMV is also responsible for antibiotic transfer genes in E. coli, Salmonella, Pseudomonas, Burkholderia. So what we are saying is that a lot of these gram-negative organisms, this OMV or MV can carry the gene responsible for antibiotic resistance. Okay, so this is how actually this report came out somewhere in 1999. If you do electron microscopy, this is a gram-negative organism, and you can see they release this vesicle. And gram-negative bacteria releasing these vesicles, MVs, and it composed of outer membrane, as I said, the outer lipid bilayer, and then it contains enzymes, LPS, toxins, and outer membrane protein. Think about what they carry. Involved in survival, pathogenesis, antibiotic resistance, antimicrobial action. And it delivers also hemolysin or phospholipase to host cells. So it's kind of like a attacking device, and at the same time it can carry antibiotic resistant marker. Everybody remember that now? So the OMV within that community, they can carry this. And there are a lot of research actually in progress. These are more recent review article 2023. So we are saying is that the bacteria releases OMV as a decoy for bacteriophages. Let's see if you understand this. Bacteriophages attack bacteria, right? So if the bacteriophage attack a bacteria, it's going to kill it, right? But bacteria want to survive. So how do I prevent attack by bacteriophages? What do you do? So for example, you are at home, right? And a bad person getting in trying to kill you. Think about it. So I found a defense strategy. So what I did, I created a plastic dummy, look like me, put it in the front door. Right? So he thought that's you, so going to attack you, right? But what I did, in order to defend myself against that attacker, I created a dummy. That's what we call dummy, right? And so dummy is standing there. So the bacteriophages think that this is the person I'm going to kill, right? But it happened to be dummy. And how it happened? Because this outer membrane, where the receptor is, where the bacteriophage is going to bind, so instead of binding the actual bacteria, it's going to bind to the vesicle. And so if the bacteriophage binding to the microvesicle, eventually preventing the bacteria from dying or getting attacked. You see this one? And that's a clever strategy. Okay, they do that. And also they carry also siderophore, which binds the iron. Okay, so it has a lot of those functions. And so the bacteriophage attack and endolysin binding. So if you read this, especially this article, you'll find out that microvesicles have a variety of role, not only protecting against some infective agents, and also can help the microbes to invade host or the pathogenic factor. Okay. So this is, I think I talked about my research in the morning about the endocytosis mechanism, and those caveoli, the microvesicles also taken up by the host cells through this type of pathway, endocytic vesicles. Whether it's a clathrin coated or caveoli, this is how we take it, and it contributes to the immune system in a complex manner, but it works fascinatingly important for bacterial infection or microbial infection or host defense. So this is another way the microvesicle forms. This is kind of, they came up with this beautiful name, OMV, OIMV, EOMV, CMV, and all of those are written here. One is, I like it most, called explosive outer membrane vesicle. Explosive means how it exploded and then created that EOMV. The bacteria exploded one side. And again, this is a recent, relatively newer article, and you can see how these microvesicles are formed. Again, they are playing important role in antibiotic resistance. They are in the organisms are in the micro environment within the biofilm, and these genes coming out of these cells from one member and then distributing these MVs within that community, then the other members within that community, they are accepting this. So the genetic exchange happening, right? Not only by bacteriophage, but also the MV, these vesicles contribute to that function. Okay, different triggers that induce membrane vesicle formation. this kind of tells you what really causing that. So the iron limitation, signaling molecules, hydrophobic compounds, antibiotics, DNA damaging agents, peptidoglycan degrading enzymes. So a lot of those factors contributing to the formation of these bubbles or vesicles from this microorganism. This is a nice cartoon illustrating how these microvesicles are formed. Got it? Okay. These are also more roles of membrane vesicles during phage infection. So that's what I was mentioning, the decoy and helping these parts to bind instead of binding to the bacteria. Okay, and that's advantage for the bacteria to survive under that condition. Okay, and these are some of these experiments I think showing that if you have microvesicles, you have antimicrobial effect is stronger. If you don't have, it's a control, and if you made that MV, you kill more microbes. So this microvesicles can be designed in a way you can target certain microorganisms. Read this review article, or this article can explain that more. So anyway, so these are the varieties of mechanisms help create that antibiotic resistance for the cells which are in the biofilm. Okay, we started with this kind of giving you an idea that with the different strategy help the microbes to become resistant to antibiotics. So those are the materials I had to talk about this antibacterial or antimicrobial resistance contributed by biofilms. Any questions? I know I said I'll finish it soon, even though it's past 4 o'clock, but we started late. So any general question or specific question? Go ahead.
Can be used for the development of any kind of antibiotic?
Definitely, you can create or design in a way that you have these molecules. You heard about liposome? Anybody? These are lipid bilayer membrane, right? You can synthetic virus, synthetic bacteria, synthetic target. So in this case, if you are trying to create a bacteriophage resistant organism, especially for lactic acid bacteria, you don't want to be attacked by viruses, bacteriophage. But yes, you can create, generate, because you have a lot of options to make it, how you want to design it or customize it. Yes, you can do that. Any other questions? Microvesicle is a fascinating area. I mean, a lot of work now in progress trying to understand how they transfer, how they contribute to pathogenesis, contribute to antibiotic resistance. Can we manipulate to our advantage to control infection? You see this? Because they may be easy to deliver, or you can construct microorganisms that can deliver certain drugs or antibiotics to kill certain organisms. Yes. Anything else?
Sir.
Go ahead.
Sir, when we talk about eukaryotes,
Yeah.
they have a complex cytoskeletal system which helps them taking stuff like membrane vesicles to the outer membrane. But we are talking about prokaryotes, the bacterial study.
No, these MVs are all coming from prokaryotes. Don't get confused. All those OMV I'm mentioning, these are all bacterial or prokaryotic.
Sir, but my question is that how these OMVs go to the outer membrane surface when they don't have a proper cytoskeleton structures like microtubules or microfilaments?
For host cell to take it?
No, like they are Yes, sir. Like bacteria is synthesizing outer membrane vesicles.
Yeah.
And they are exporting it to the outer membrane. They are synthesizing inside the bacteria.
No. Okay, so let's go back this one. Look at this structure. Right. So this was what we're saying is that number of ways they make it, bacteria. With that, it's like an explosive. Whatever the treatment we had, so for example, endolysin. You heard about endolysin? This is the phage protein which causes the lysis, actual enzyme, break it down. And so if the endolysin is there, so a lot of this nuclear material packed in this outer membrane vesicle and comes out as like a balloon or ball, okay? And that's what is carrying this genetic material inside. So there is a number of mechanisms, that's what I was trying to explain, that these are the factors, and it's kind of like a blabbing, it comes out. Not necessarily sometimes killing the bacteria. For example, if it forms like this balloon and you clip off, that comes off. This is how they are forming it. The different mechanisms are involved forming these microvesicles that come out of the microorganism. That's the question, right?
Yes, sir. Got it.
Yeah. So now you can see this. Sometimes the bacteria has to be totally lysed in order to make the small fragments, and then that can be taken up by other microbes, or the gene can be transferred under that condition. And for eukaryotes means that can cause a serious disease in humans, because each of those components taken up by the macrophage or phagocytic cells, and then they can cause damage to those immune cells, right? Because they may be carrying toxins, they may be carrying some other molecules which inhibit their growth, and that way they can also suppress immune system. But the immune system also try to overcome that and try to provide protection against those type of incoming toxin, for example, or phospholipase, all of these factors. It works both ways, right? Okay. Any other question? So I think the picture I think I showed you in the beginning, that's kind of fascinating. It's like very old, but you can see the under electron microscope, you can see this E. coli cell. I think this is E. coli. I remember, but this fly is bugging me. So they are forming these vesicles, and you can see clearly coming out of the cell. The fly is bugging me, right? Somebody said bug. Bug. What is the product he was mentioning? Bug. Huh? Good bug product. I don't know. This should not use the word bug with a good or food. Anyway, anything else? No, I know it's already late, so it's 4:15. If we don't have any more questions, we'll see you tomorrow morning, and we'll start with the control of biofilm, right? I think that's the scheduled lecture. We'll start with that, then we'll try to wrap up most of the lectures tomorrow,